Hello, everybody. Welcome to the stream. Today we have very exciting stream for you all today. It's about genetics. Hello, A. Welcome. Today we are going to be talking about something that I was somewhat actually dreading talking about because I don't fancy myself to be a geneticist, but not too long ago, I came up with an angle that allowed me to talk about this topic in a way that I think I'm really excited to share with you all. And that's going to be taking a look at one of the more genetically mysterious conditions, and that would be schizophrenia. So the plan for today is to take you from Genetics 101 all the way to trying to comprehend a very difficult to understand disease. And I think that process is going to be very fun. Hello, General Mustache. Hello, Silly Ciliate and Vincent Peterson. Welcome to the stream. Before we begin with this stream proper, I just wanted to shout out, wanted to congratulate fellow V-Theory member Sidere. Sidere is another VTuber in this group that I'm in called V-Theorem. Bunch of STEM-based VTubers, and she recently won an award or a recognition for her work on Sidere channel, which is very exciting. Although, being the ever-competitive person I am, I cannot wait for my turn to be able to make that kind of same announcement. I'm very proud and happy that she's won this award, but I also can't wait for my to get my own grubby little hands on something similar. So that's very exciting. And also, before we get into genetics, let's talk a little bit about some of the stuff that's coming up for this channel in particular. Hello, Brandon. Welcome to the stream. Today, we're going to be doing MolBio 101, talking about genetics. Next week, we are going to be chatting with another VTuber, Resta Makurabe, 8 to 3, 2 p.m. EST on bioinformatics. Right, bioinformatics. So today we are going to be doing genetics, and then if I said I was a little uncomfortable with genetics, I am really uncomfortable with programming. But I'm very lucky to have a VTuber who has done a lot of that in a professional capacity. So that's going to be very exciting because instead of me putting together the lecture, it's actually going to be Resta who does. And so I'm going to be able to sit back and just enjoy what she has to say along with the rest of you, which that would be very exciting. And then we've got my 1-year anniversary stream coming up on the 11th at 3 p.m. EST. It's kind of crazy that I've been making content for a year. And so I wanted to do a little special little birthday celebration for that day, which I'm really excited for. At some point, I will have a members gaming kind of thing, especially because the school year is coming up and I want to get one last hurrah of goofing off playing video games before class officially starts back up. And along the same lines, I also want to do a little bit of a teaching and learning meta stream where I talk about sort of the ongoing research and how teachers can best tune their content for students, as well as what students can do to more fully engage with teachers. And I'll probably be collecting some feedback from the audience, you all, about what your school experiences have been like. And then finally, something that I'm trying to plan for at some time in September, it's this paper on the brain bacteriome/virome. So that is going to be a members suggested stream, so that if you don't know, if you join membership at the tier of T-cell or higher, you get access or the ability to suggest content for me to cover, which would be a pretty fun way to be able to talk about stuff that I normally wouldn't talk about. And so that's a pretty interesting place to be in. All right, with that out of the way, let's talk a little bit about molecular biology, why don't we? So let's just get right into it. So one of the first entries in this Molecular Biology 101 series is this stream on the molecule DNA. So I did this a while back, and I'm very proud of this of this title. But you actually don't have to have seen that stream in order to appreciate genetics. In fact, the founding of modern genetics was actually done without knowledge of DNA, and in fact, without knowledge of evolution. We're going to see how the brilliant scientist Gregor Mendel put together the basics of what we know now to be the field of genetics. Okay. So you've seen this plenty of times before, especially if you've consumed my content for some time. This is the central dogma of biology. Of course, we go from DNA to RNA to protein. The process from DNA to RNA is transcription. The process from RNA to protein is translation, right? I've mentioned this so often. I'm thinking that if I ever release merch, one day, this is definitely going to be something that I have to think about in terms of how this would translate to a shirt or something, because this is truly central to biology, ergo central dogma, right? But actually, genetics is oof, the word art. What do you mean? I love the word art. The thing is, genetics is freaking complicated. All right, genetics is really, really complicated, and it's part of why I was kind of procrastinating on putting this stream together. And that's not to like poo-poo the people who like genetics, but even people who like genetics probably can admit that genetics is very complicated. And so to give you an idea, even though the central dogma can be summarized in 2 arrows, there's this central dogma extended universe. I'm going to move chat for a second so that we can sort of see the other stuff that is also sort of around the central dogma, right? So the central dogma describes the flow of information, and this extended universe more describes interactions between the different molecules. And I'm sure this isn't comprehensive, right? So not only do we have transcription and translation, we also have RNAs regulating each other. We have proteins making other biomolecules like lipids. We have the entire fact that we intake biomolecules from the environment through nutrition and stuff like that, as well as exposure in our environment. We have the fact that proteins also regulate gene expression, et cetera, et cetera. So even though the central dogma is a really good rule to keep in mind, the lived reality of how organisms are built is really, really complicated. I reject the central dogma because I intend to defeat genetics and become taller. My mom actually signed me up for swimming lessons because part of her desire was to lengthen my body. I was, I guess I won't talk too much about body image. It's not the stream for that kind of thing, but she had this belief that because I was in a low gravity kind of situation that would elongate my spine. I don't think that that's how that works, but that's certainly one attempt at rejecting the central dogma. Okay, so where do we start with talking about genetics? One very, 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 very lucky observation and one very, very, very persistent scientist, Gregor Mendel. If you've ever taken a biology class, you might recognize this name. Hello, Seth, welcome to the stream. Definitely working on making 10,000 more, don't you worry. So Gregor Mendel, the father of modern genetics, lived and died in obscurity, which is very tragic considering how important his work was. So Gregor Mendel was a priest, I believe, and this priest was doing a lot of work in understanding the inheritance patterns of plants, specifically the pea plant. And so what strikes me about Gregor Mendel's story is that he had no idea about evolution, right? Darwin and Mendel were of different time periods. Certainly Gregor Mendel had no idea about DNA or the molecule of inheritance. He just noticed, hey, when you breed certain varieties of pea plants, they pass on their traits in certain patterns. And so what Gregor Mendel did was he spent a lot of time breeding plants for certain traits and making sure that they were very pure, and then setting them up in specific crosses to understand how these traits were passed down. So for years, Mendel worked on this project, breeding pea plants, making sure that they were pure for this kind of observable trait, and then examining how this set of traits was passed down from generation to generation. So these traits included seed shape, flower color, seed, coat tint, pod shape, unripe pod color, flower location, plant height, and just seeing what patterns these traits were inherited by, without, again, any knowledge of evolution of DNA. Okay, so before we get into the findings that Gregor Mendel had, we first have to define a couple of terms. And so if you've ever taken a biology class, you will be familiar with these terms. Traits, just another word for phenotype. So when we talk about phenotype, we're talking about something that you can measure, an observable trait. So eye color is an example of phenotype, as well as all 7 of these. They're all phenotypes, right? The genes that underlie them is the genotype. And now Mendel would not have known anything about genes or genotypes, but for our modern understanding of genetics, that's what we'll work with, right? Okay, so when he was observing the inheritance patterns of these pea plants, he came up with several laws, and these laws govern the way that these traits would be inherited, and so we called them the rules for Mendelian inheritance. So some of these traits, and Mendel was actually very wise. He didn't think that he had cracked genetics, right? He believed that there were certain traits that behaved in this very defined way, and so he described 3 of these laws: the law of dominance and uniformity, the law of segregation, and the law of independent assortment. And all of these laws hold true for a very specific subsect of the genome. And so we'll get into what each of these laws actually govern, I should say. So the first law is the law of dominance, right? So for example, if you had 2 plants and one had a certain phenotype and the other had a different phenotype for a Mendelian trait, if you were to breed the 2 and one of them had the dominant trait, then all of the following generation would have the trait of the dominant gene, right? So white here would be recessive, and then orange-red here would be dominant. And so if the red trait is the dominant trait, then all of the children of that generation should be red, right? This is pretty simple to understand, and indeed, in your Biology 101, the first biology class that you'll likely take, will explain these rules and then make you do a bunch of boxes. What are they called? I don't remember. The crosses, which we're about to do right here, right? Oh, okay. So if capital R is dominant and small r is recessive, just for a bit of audience participation, oh, actually, I just printed the answers. Anyway, it's not that hard to understand, right? So we know now that these pea plants are diploid, meaning that they have 2 copies of every gene, and each one of those copies will segregate independently of one another. And so what that basically means is that you have 2 copies of your genes, although one copy can vary slightly from the other copy. And the important thing is that when genes are passed on, the cells that will become the eggs and the sperm or sperm equivalent will carry one of those copies, right? And so if you have a pure white phenotype here, both of these recessive traits will segregate. Same with the dominant trait, and so you get this split. So this red parent will donate 2 capital R's to red traits, and then the white parent will donate 2 lowercase traits. And so all of their children will be a combination of the uppercase R and the lowercase r, a combination of dominant and recessive traits, and therefore, because of the law of dominance, all of them become red, right? So this is recap. And this is indeed what struck me most about learning genetics for the first time, because I thought, hey, that's pretty easy to understand. Genetics must be pretty simple if these are the rules that we abide by. But as we'll see in just a little bit, it gets much, much more complicated. So if we apply the rules of dominance and the rules of segregation to the next generation, if you were to cross these plants that have both one red trait and one white trait, then we see that if we split those alleles like we have to in the law of segregation, that 1/4 of the offspring will get 2 copies of red, 1/4 will get 2 copies of white, and then 50% will get 1 copy of each. And so you get this 3-to-1 phenotype pattern, but the genotype pattern is 1-to-1-to-2, right? So hopefully that's not too difficult to understand, or this is just a refresher for Mendel's basic laws. But I'm going to pause here and make sure that we are comfortable with this idea of Mendelian inheritance. So I'm going to go ahead and put up a poll with Mendelian inheritance. And I know I kind of zoomed by that, but I kind of assume that my audience has had at least one biology class, but it's totally okay if you want me to take another crack at explaining Mendelian inheritance. All right, so we're rolling in, yeah. The ideas of dominance and recessive genes, they are deceptively simple, as in that if you sort of take the assumptions that Mendel has for you, then the resulting conclusions are relatively straightforward. Where's the 4x4 Punnett square? Ooh, we are getting that, Toxo. See, the 2x2 isn't bad, but we're going to get into the 4x4. The 4x4 is a little nastier. Do I have any advice for someone going to AP Bio? Yeah. Don't procrastinate. The thing about biology is that in order to have fun in biology, you need to know everything before you start having fun, and it can be quite discouraging because it can be kind of hard to force yourself to learn everything. But if you want to do biology professionally, you gotta do it. There's no easy way around memorization. I would say that if you have the ability to access practice tests, that you should be doing those constantly. There's no better way to study than to simulate a test environment if what you're going to be graded on is testing. So that's my quick spiel for how to be an effective student. Okay, so Toxo teased this 4x4, and we're going to get to that. But we're actually going to cheat through it because I'm not going to make you do it. Because these are kind of tough. And actually, there was a Neopets puzzle not too long ago that basically made you do this kind of thing. I don't know if if I even have audience members old enough to have played Neopets or even be aware of that puzzle. But the thing about Mendelian inheritance is that there's this third law called the law of independent assortment, and that's if you are comparing different genes for Mendelian traits, those traits will segregate independently, and that's because some of these genes just happen to lie on different chromosomes. So let's see if I can actually get I can get my cursor to show up. Wonderful. Okay. This is big because I had trouble with this last stream. Neopets puzzle solving stream. It took me way too long. It was embarrassing how long it took for me to solve that puzzle. Okay, so if we look at the chromosomes in these gametes, then we see that R and Y, these traits here, they're on different chromosomes. So the way that they separate has nothing to do with each other. So when they form their eggs, you're going to get all sorts of different combinations between the gene R and the gene Y and their various different combinations. So if we're testing for two traits, right, because the 2x2 case is just for a gene, this is examining the relationship between inheritance of different traits, not the same gene. Right, so not only are we looking at color, we're also looking at pod shape and understanding that in the law of independent assortment, that the segregation of the pod shape and the pod color are independent. They don't affect one another. That's why you have so much different variety, different combinations up here in this generation. It's also why you get this horrible 4x4. So if we're looking at mixing multiple different traits, you can often get many, many different varieties depending on how those individual traits are inherited. Does that kind of make sense? How deep do Punnett squares go? I've only done 2 genes. It's not practical to do more. I think the only reason to do Punnett squares on 2x2, and again, I'm not a professional geneticist, but in terms of when I was in graduate school, you only had to know how to do a 4x4 because it illustrates this law of independent assortment. This tool, I think, again, for a non-geneticist, just for your stock biologist, this tool is just to remind you that genes on different chromosomes don't affect the segregation of the other gene. Although, as we're going to talk a little bit about in non-Mendelian inheritance, that's not necessarily the case, which is why this is only valid for Mendel stuff. Abigail, keep all your notes from AP Bio. I still use them halfway through my undergraduate career. That's only if you are the type to refer to notes. I actually am a bad note taker and a bad note reader. So I actually end up not using my own personal notes. Like, you'll find your own learning style, but the empirically tested solution that works for the most diverse learners, from my understanding, from what I remember learning when I was studying teaching, like formally as an academic study, is test taking. That doesn't mean that note taking is bad. It just means that for bang for your buck practice tests, that's the way to go. I miss AP Bio. I don't. My memory is really bad. I actually did not very well in AP Bio, but it just goes to show that if you love something enough, even if you're bad at it, you'll find a way to eventually get good at it. I use other people's notes. I just went off of lectures and my textbook, which was highly dependent on the quality of stuff, the material that I would get. I don't always recommend it depending on who's teaching you. Okay, so the law of independent assortment, different traits don't affect the segregation of other traits in Mendel's context. If we're talking about non-Mendelian inheritance, is a different story. Oh, and welcome, Icosahedron. Welcome to the stream. Didn't say hi to you yet. Okay. I keep on harping on this, that all of these rules are just for Mendelian traits. And so why is that? Again, because when I was in high school, I underestimated how difficult genetics would get because I thought Mendelian traits were the majority of how genetics worked. And in reality, Mendel actually found a very, very lucky system. He was very, very lucky and very, very disciplined to have picked the pea plant because the pea plant behaves in a way that is easily understandable. So these traits, these laws that describe Mendelian traits, it's like incredible how Mendel just happened to pick the perfect organism to study these in because the inheritance patterns are so simple. Because, as I'm about to get to, the majority of life does not abide by these simple rules, for better or for worse. So how many traits are purely Mendelian? There are many diseases that have Mendelian inheritance. Like if things go wrong, there's a decent chunk of them that go wrong because of some Mendelian inheritance patterns. But basic body stuff tends to be quite complex, like height. Height is a case where it's not just one gene that you get the tall gene and you're tall. Unfortunately, that's not, or fortunately, it's not as easy as that, right? Often genes are much more complicated than just dominant and recessive. Except weirdly, earwax, apparently, and this is something that I just learned while prepping for this stream. If you have the wet earwax trait is actually dominant, and then the dry earwax trait is actually recessive. So apart from earwax, most regular body stuff is not going to be fit in this category, which I think is pretty interesting. So to illustrate this, I have this spectrum, and like many things in biology, the complexity of genetic inheritance is also a spectrum, especially when you start thinking about molecules, because I think chemistry is really guilty of this problem, but biology is somewhat guilty of this too, is that the stuff that you learn in your first chemistry class often gets overturned by the time you're in undergraduate, and definitely by the time you're in graduate school. And there's some of that in biology, although most of the biology you learn early on is still true, or good enough, I should say. But in terms of how I learned genetics, there definitely should have been a higher emphasis on the fact that Mendelian traits are the minority here. So there are some traits that are Mendelian. So male pattern baldness, that is one of the traits that is Mendelian, and it is quite straightforward. So if you want to predict if you will go bald, you should look towards your mother's father, and then that will inform you about what direction your hair will go as you age. Color blindness, that is another Mendelian trait, and the gene for sickle cell is also Mendelian, so dominant, recessive, independently assorted, etc., etc. But as you can tell by the gap here, most of your genes are very complex and non-Mendelian. All right, so next to, or in my view, what is the next thing down from simple Mendelian inheritance is this idea of incomplete dominance. Does male pattern baldness affect neutrophils? I mean, you say that, but we're, as a joke, but as we'll talk about in just a little bit when we get to epistasis, the question of if genes affect other genes is a huge headache. But we're not there yet. We'll talk about epistasis in just a little bit. Let's go simple first. Incomplete dominance, as you see here in this graphic, say you have a red colored petals and you have a plant with white colored petals. If all of their offspring have purple colored petals, then this trait is said to be incompletely dominant. Mice fur color is Mendelian. That's interesting as well. I was not aware, especially considering the mice that I use are always inbred, and so we don't really get to see inheritance patterns of fur. But incomplete dominance, so incomplete dominance and codominance are slightly different. So incomplete dominance is defined by a quantitative interaction between the quote-unquote dominant and the quote-unquote recessive trait. So quantitative, so numerical changes. So if red is 100% red and white is 0% white, then purple in this particular case is like 50% red, right? So there's a numerical interaction here. So that's important to keep in mind for incomplete dominance. Codominance is when both traits are both expressed equally. So in this particular flower, we see both the phenotype of red pigmentation and white pigmentation. So both of these traits exist at the same time. So this is codominance, right? So you can already see that this idea of dominance and recessive doesn't quite explain genetics very completely, because you do get these interactions in gene product. And here we go. This is epistasis. So in this particular graph, this is a graph describing baldness. I don't actually know if this is true. I didn't really look into whether or not baldness is inherited in this way, but it looks like we have on the top here some genes for hair color, but both of these genes for hair color are overwritten by the baldness traits. So you won't get the expression of blonde hair or red hair on your head if you're bald, right? So the bald trait epistatically affects the phenotype. You still have the gene for blonde hair or red hair, but you never see that because the gene for baldness is covering that up, right? So that's epistasis, one gene affecting others. And it's going to be the bane of any biology student's life when you get to that, because it's complicated to figure out. And then we have genes that are multi-allelic, so genes in which different varieties of that gene will have different effects, as well as polygenic, so many genes contributing to one phenotype, right? So this is the two branches. One gene, many effects, one phenotype. You can see them as like branching out and then branching in. And so most of your traits are inherited not as Mendelian, simple recessive and dominant traits, but some messy, terrible combination of everything on the right-hand side of this graph. But it's also, I think, why geneticists like this kind of thing, because it is that nasty and that complex. So here's a really fun activity I thought we would do. Let's hypothesize some molecular mechanisms, right? Because I'm a very molecular kind of guy. When it comes to big data and genetics, that kind of stuff doesn't excite me all that much. What I am excited by is thinking about how molecules lie underneath inheritance patterns, because geneticists, this is what they care about, how parents or how children inherit traits from their parents. That's primarily what they care about. But a molecular cell biologist wants to know underneath that inheritance pattern what is going on underneath the hood. So the majority of you were comfortable with simple dominant and simple recessive. And so as an example of this activity, I'll just do those and pitch hypothesized mechanisms of those inheritance patterns, right? So if you have a simple dominant trait, one reason why that trait could be dominant is that version A makes a toxic protein, right? So this is, I should have looked it up before the stream, but I believe this is the inheritance pattern for Huntington's disease, right? So Huntington's disease is I believe it's an autosomal dominant, although someone can fact check me on that in the chat, disease that you inherit, where if you get one copy of a Huntington gene that has too many repeats at the end of the protein, CAG repeats, that Huntington protein can kind of aggregate. And if you watched my prion video, you would know aggregation of protein in neurons, it's a very bad thing to happen. So one way that a gene could be dominant is that the dominant version of that gene causes something toxic, and that toxicity can't be fixed by the healthy copy, right? And so on the other side, a simple recessive trait would be something like, this should be one copy of version A is not enough to be healthy, sorry. So this thing that I've written up here, that's a mistake. That should be one copy of version A is enough to be healthy. And so chat, I want to challenge you to think of a way for incomplete dominance to happen. And if you're a biology student, let's just give the non-biologist in chat a little bit of a head start. Yes, I'm right. Huntington is caused by too many polyQ repeats. Yeah, okay, yeah. I think polyQ, I think Q is coded by CAG because that's what I remember as being the inheritance for Huntington. But yeah, chat, let's brainstorm. This is the fun part of being a molecular biologist. For a trait that is incompletely dominant, what do you think is happening to the proteins? Like, why would proteins, how could proteins give rise to a trait that is incompletely dominant? I kind of gave you a hint when I talked about flower color before. And again, remember that incomplete dominance and codominance are different because incomplete dominance has a quantitative, a numerical interaction between one another, where it's not quite one version of the gene and not quite the other version of the gene. Give you a chance to think that over because it's an interesting question, right? Even if we just sit here for a bit and just ponder, when you get the answer, it'd be maybe more satisfying to think about, right? So I'll give the non-biologist, like, I don't know, another 10-ish seconds, and then I'll ask the biologists in chat for what they think. But chat, make a stab at it. If you are wanting to be a biologist, this is a perfect time to take a stab at this and make mistakes where there's no penalty, right? If you're wrong here, it doesn't really matter. And forcing yourself to come up with a answer is a good brain workout, and it'll make you less afraid to think. Because I think for me, when I was growing up, I grew up with so many little geniuses around me, and I always felt bad about participating because I would fear that I would look stupid next to my classmates, when in reality, that really does handicap you when you start to push yourself as a learner. Forcing yourself to learn actively in situations is statistically proven to result in better learning outcomes. Quantity of proteins, that's a guess from A, so that is interesting. That could work. Yeah, quantity of proteins. So let's see. So let's hypothesize one version of a gene could be coding for, let's say, a genetic regulatory element that asks a protein to be made more, and then one version codes for one that isn't quite as strong, and so maybe you get one version or one copy of that gene that is made at a very high level, and then one version that isn't made as much. That's a very good hypothesis. Quantity of protein definitely could be one particular mechanism. All right, with that, biologists, if we have biologists in the chat, I will take answers from biologists if you know the correct answer, or at least the answer that I'm pushing, because A actually brings up an answer that I was not considering, but I believe that totally counts unless it's a gene that's always coded by one particular copy, which I don't know how many of those there are. Yeah. So biologists in chat. Or have I stumped the biologists in chat too? That would be very interesting. Misfolding of proteins not being effective at expressing the gene. Yeah. So that's actually a very specific hypothesis, Brandon. That's very specific. I went with very general descriptions, but that certainly fits what I would say as a mechanism of incomplete dominance. So I'll give that away here. Okay, so what did I say for incomplete dominance? Let's say version A of the gene has 100% function, and version B is totally non-functional for whatever function this protein needs to have for this particular phenotype. So if you get one copy of version A and one copy of version B, then you end up with something that is 50% functional. And that makes sense given what we see here with the flower colors, right? If 2 copies of red gives you red, and no copies of red gives you white, if you get 1 copy of red, maybe that gives you purple, right? I'm listening to this as I'm doing PCR. Oh, PCR. I'm a geneticist. Awesome. So I'm glad to see you talking about my favorite topics. I learned this in genetics, and the answer, I think, is sort of nicely intuitive. Yeah, there's a lot that's really fun about connecting molecular mechanism to genetics, because I think what I'd struggled with learning about genetics in grad school is the disconnect between inheritance patterns and living biology. I'm always more of a fan of looking at organisms rather than looking at math. Doing PCR on a Saturday, though, that sucks. Okay, so what about codominance? Codominance. So again, we'll give our non-biologist a chance to answer. So both versions of this gene are equally expressed. So one gene does not cover the expression of the other. You get two versions, a double expression of a certain type of trait. So and I just want to address, Brandon, your answer of misfolding proteins not being effective. That's basically what I'm saying here too, right? Like one version, if you get a mutation, maybe that version or that copy is not effective, but maybe it's not toxic, right? If version B had 0% function and was toxic, it would probably look like simple dominance. But if it's just like garbage, if it just misfolds and it can't operate, then you only have half of the healthy version, and maybe you get an intermediate, not quite dominant, not quite recessive. So codominance, two expressions, one gene, two different phenotypes, both expressed. How might this happen? Or, alternatively, does anybody in chat know of a real-world example of codominance? And it's something that we should all actually be quite familiar with. Welcome, Kuro. Welcome. We're talking about genetics. Welcome. Yeah, finally caught a stream. I know. You also stream at like crazy times, but I'm a morning person, so I end up being able to catch your streams. I actually have no idea what time zone you stream in because it's always really early where I'm at. I'm in C. elegans biologist, and they can be rather stochastic in how fast they can age, especially with knockouts. True. I learned a lot about C. elegans when I was learning about longevity, although I ended up not going into longevity as a science, but super interesting how they're there. Heterochromia as an example. Oh, yeah, I don't know if heterochromia counts as codominant. Somebody might have to look that up for me. And the reason why I say that is because I'm not sure eye color is one gene. It might be a couple, but codominance might be a part of the story of eye color. That's really good. So there's another example. Blood types. Yes, thank you, Kuro. Yes, blood types, A and B. You can have blood type A, where your blood cells have that antigen receptor, and then blood type B. You can also have AB, right? So that's codominance. Eye color is multi-allelic. Yeah. So that's why I was a little nervous about calling eye color strictly codominant, because it could be codominant and a whole bunch of other things. So I think eye color is a bit too complicated. But that's a good example. So how might codominance, what might underlie codominance molecularly? Right? Molecularly. Eye color 16 genes. Yeah, okay, good. I don't want to like say eye color is codominant if it's 16 genes. That's definitely really complicated. I have no idea what the conversation is about right now. We're talking about molecular mechanism for inheritance patterns, because what I didn't like learning about in genetics when I first learned it is the disconnect from real biology, right? We learn these rules and then we learn about flower color, but there's like, it's like not satisfying to have a full picture. So we're trying to fill out that picture and analyzing what is underneath the expression of phenotype, and that's molecules, right? So how might something like codominance be expressed with proteins? And Kuro brings up an excellent point. Blood type actually gives away that answer, right? So I'm actually going to fill this one out for you, because blood type is exactly what I'm referencing here. Okay, so if we look at blood type, version A is function X, version B has function Y, and if you get one copy of each, you have XY, right? So if mom has two copies of blood type A, dad has two copies of blood type B, you will always get blood type AB. So A doesn't cover B, B doesn't cover A, you are AB. So that's codominance, right? So each individual version of that gene might have their own function. And so that's definitely true of blood type. So I hope this is making some sense in terms of where we're at. The molecular mechanisms behind inheritance. Okay. Epistasis. This one's tough. Okay, epistasis is tough. We love O- universal donor and AB universal receiver. True. don't forget to donate blood. Kind of off topic. No worries, no worries. I have donated blood multiple times. I've actually also donated platelets, I believe, which is an interesting experience. If you've never donated platelets, it's certainly something to try at least once in your life, just for the unique sensory experience. But yes, donating blood is always a good thing to do. And in some places you can get recompensated for it, which is fun. So epistasis is a genetic interaction where one gene can interact with another gene. Platelet donations are different. Yeah, so they actually take your blood and then they spin down the blood and separate your blood from the platelets, only collect the platelets, and then you get your blood back. And that feeling of receiving your blood back is really weird. You have experience with donating platelets. It takes ages. Yeah, it's like 2 hours. You do have to chunk out a whole portion of your day. But actually, if you donate platelets, you actually do get monetary recompensation, and it's more than red blood. So if you I did it because I wanted to, I don't remember what it is I wanted to buy, but I was like, oh, to justify this purchase that is a little outside of my comfort zone, I'm going to donate platelets and then buy the purchase. I don't remember what it was, but yeah, convinced myself to do it. And getting back your cold blood, it's definitely interesting, and I do recommend it just for it being interesting. And if you find that you can tolerate that experience, then you would be contributing to the betterment of a lot of people's lives, which would be very interesting. Money, I can get money by donating things. Yeah. Donate platelet chance for a GPU. I don't know how long you would have to do that for a GPU. Maybe over the course of years. Do they use anticoagulation factors in the meantime when you donate platelets? I actually don't know too much about the underlying science behind platelet donation. Okay, sorry. Back to epistasis, because this is something that I think will unlock thinking about how complicated genetics are, because incomplete dominance and codominance, these are just slightly spicier versions of Mendelian inheritance, right? It's a little spicier than just dominant and recessive, but epistasis is way too spicy. It is the like bulldog 3x spicy chicken spicy. So epistasis is when one gene masks or affects or interacts with other genes and can affect their expression. So this one is pretty tough, chat. And I will be really proud if anybody, especially if you're a non-biologist, if you can figure out a way for molecules, because again, all genetics is setting up molecules, how proteins or gene products can result in an epistatic relationship, right? One gene affecting the expression of the other. Baldness, masking, blonde hair or red hair. That's epistasis. If you get the bald gene, it doesn't matter. If you get the blonde hair gene or the red hair gene, you don't see that hair color. So, chat, any guesses? And this will really push you, I think, to think creatively, which is what science is so fun. What makes science fun? Like, I think the best part of graduate school, I think for me, was sitting at my whiteboard, like, math equations, although I didn't do any math in graduate school, but like, diagrams and math flowing in the background, that kind of thinking and guessing really was the fun part for me in graduate school. Don't ask me for biology trivia. I just graduated middle school. It's not biology trivia. It's asking you to generate a hypothesis, right? Generating hypothesis is the best. The bad thing about science is when you actually have to test that hypothesis. That's difficult. That's really hard, and you can spend years doing that. But thinking about a hypothesis, that's goated. That's the best part, man. That's so fun. It's just thinking about ways that reality can be real. But actually testing it is a lot harder. So ASA molecule turns off gene. Yeah, that's definitely an answer, and we'll talk about that in just a little second. Protein doesn't let the phenotype to even appear. Everyone has the ability to come up with hypotheses. Exactly. That's the whole fun of these streams, is that, again, if you're wrong, this is a very low stakes environment to be wrong in, and it's a way for you to practice thinking, which is really important to me. Also, I don't think I've said hi to you, Morjuice. Welcome in. Protein doesn't let the phenotype to even appear is exactly how I would describe that. But actually, A's language is a little more close to how I would phrase this as an epistatic relationship. So this is certainly not the only way that epistasis can happen. Can hyperactivating proteins also be a thing? Ooh, Kuro, that's an interesting answer. Okay, I will first go through my answer, and then we'll brainstorm ways that Kuro's answer can be true. Kuro's kind of cheating a bit because Kuro is studying medicine or preparing to study for medicine, so quite a bit more familiar with this material. So let's say the presence of protein C blocks gene ampersand. So this gene, this hypothetical gene that we've been referencing, let's say that there's some other gene in the genome that makes the protein C that stops gene ampersand. So let's say gene ampersand makes a protein that, for example, gives you hairy arms. I don't know. And then protein C, maybe that protein binds the hairy arm protein and prevents it from getting to the skin. Something like that. That interaction could be very possible. If I lost the gene for producing skin, then how can I know that I also have alopecia? Sorry about that. Alopecia is that the hair loss one? I'm unaware. Forgive my ignorance. I'm a med student. They don't teach us this in genetics. That is interesting to me. Maybe I am underestimating how different genetics education is across different school systems, because this was stuff that we were taught but never, I think, given the full appreciation for. So let's revisit Kuro's idea, because I think that's really interesting. Can hyperactivating proteins also be a thing? So let's say that gene ampersand makes a protein product, and let's say protein C is a transcription factor. So transcription factors are proteins that activate genes or help genes be made into mRNA, which can then be made into protein. So maybe, just thinking hypothetically, maybe there's a protein C that is a hyperactivating transcription factor, one that really loves binding to that region of the DNA, and maybe that relationship can overcome when that gene is supposed to be turned off. I think, Kuro, your example can work as epistasis, but it takes a real geneticist. I don't know if Aiden is still out there. I don't know if Aiden would agree that that counts as epistasis. But again, these things are all on a spectrum. They're categories that we label in order for us to better think about problems, not necessarily hard truths underneath. There's a particular example of epistasis that I learned about in my genetics class. I'm biting my tongue in case you've mentioned it or have already. I can't remember off the top of my head what good examples of epistasis are in humans, at the very least. Although I think the ones that we learned about were mostly in like bacteria or yeast and things like that. I don't think those are as interesting for a general audience to think about. Outcompeting a protein. Yeah, so if Yes, that's a great example, General Mustache. So let's think about what case that would be. So say, for example, you have two proteins and they both bind a receptor. And let's say that when protein A binds, you have a cell dividing signal. And then if protein B binds, you have a apoptosis signal. So one protein binds the receptor, it says divide. The other protein binds the receptor, it says destroy yourself. And so an epistatic relationship could be that a mutation of protein B binds that receptor so tightly that you no longer get that other cell divide signal coming from that other gene. So even though you have that gene for cell division, this stronger protein masks that phenotype. That's a great solid example of how an epistatic relationship could work. So you can imagine, again, I hope we're coming to an understanding that Mendelian genetics is a sliver of genetics. It is the easiest way for you to start thinking about genetics, but it's actually not accurate to how the body works across this scope, right? The body is much more complicated than dominant and recessive. Okay, multi-allelic and polygenic. This one, Genetics makes my brain hurty. Yeah, and that's why I'm not a geneticist. I like thinking about molecules. When different molecules bump into one another, that's way more easy for me to understand than how all of this works. I had to do quite a bit of studying just to remind myself how all of these things function. So multi-allelic polygenic, I lump these two together even though these are very complicated and certainly very different. For me, for this intro kind of stream, they're the same. They're just really complicated situations. So I'll go ahead and fill this one in. So let's say gene ampersand makes a protein important for different things. So one protein, many functions, or many genes contribute to gene and phenotype. So chat, what, if any, and don't say schizophrenia because that's the topic of today's stream, but do you know of a condition or disease that is polygenic, so many genes contributing to one phenotype? I think all this is cool and I'm confused. I will understand it once I graduate high school, but I'm still trying my best to learn. I mean, trying is such an important part of learning, and failing is also a very important part of learning. So, you know, if you are coming up to hurdles in your understanding, don't worry. I find it so amazing that this is a regular feature of this channel, that the number of stream viewers that are either in middle school or freshly out of middle school is just mind-boggling to me. Like, like my content is for high school and up. So if you're confused, that's normal. It's very normal. Once you take your first high school biology class, I believe all of my content will make perfect sense to you. Autism and rheumatoid arthritis, autism, ADHD, Tourette's, skin color. Yeah, definitely. All of these. Actually, I don't really know about rheumatoid arthritis because I don't know a lot about rheumatoid arthritis. But yeah, these are all conditions that have heritable characteristics, and the fact that we haven't like found the autism gene is indicative of the fact that it is likely a very complicated story behind the genetics of autism and similar conditions. Breast cancer, that is a more complicated situation that doesn't fit neatly into this category. So I hope you as stream viewers start to appreciate how much I'm hedging. Because here's the thing, and I'll eventually do a whole stream on this, but I can't make conclusions for science in general, but biology is so messy and complicated that our labels for certain things are more of are learning and teaching tools and not necessarily hard categories. So breast cancer, would I call that polygenic? Yes. But would I also call that, it's not one that I would typically think of as polygenic, because actually, I don't know. I don't want to speak too much on breast cancer because I'm not fully read up on it. I guess I should pose this question to chat if anyone who is more knowledgeable about breast cancer can inform me, because here I am admitting the limit of my knowledge, right? Is breast cancer always polygenic, or is something like a BRCA mutation sufficient? I feel like you're right in that breast cancer is polygenic. Because BRCA is such a prominent mutation in breast cancer, I just don't remember if BRCA is sufficient or if it's one of the genes that is typically in breast cancer. If you're in middle school and you're sitting here and participating in molecular biology stream, you are definitely ahead of your peers. Yes, exactly. I'm in A-level, and the stuff I've learned from your videos has made me so much more of the content we learn a lot easier. That's great. I love to hear that. Bipolar disorder. I would lump a lot of that as polygenic, yes, just because we don't know. Actually, I struggled with bipolar as well, because not that I personally struggle with bipolar. I don't know enough about bipolar to confidently say that that's polygenic, because I don't know how much of that is influenced by environment. But certainly these are all great candidates. I'm thinking of one that I'm surprised hasn't come up yet. The one that I've been thinking of is diabetes type 2. To me, that is the classic polygenic disease/condition, because that has some degree of heritability, but we don't really know what's going on behind the scenes. Microcephaly has 28 genes potentially linked to it. That is a lot of genes. BRCA is not always sufficient. Thank you. Okay, so yeah, so breast cancer would be polygenic if, or strictly polygenic if BRCA is not sufficient. Thank you, General Mustache. I feel like cancer in general is a multi-headed path. That's true. Yeah. I'm not a cancer biologist, but that is ringing bells that cancer is generally multifactorial. Yes. Thank you. You are right, Toxo. Going to cheat for a second because this is my field of study. Idiopathic Parkinson's and Alzheimer's to an extent. Yes. Oh, Alzheimer's is another big one that I really think a lot about in terms of polygenic. All great answers. You put us in the psychological mindset with schizophrenia. That's true. Yeah, that's really my bad then. Needs multiple mutations to ruin the genetic safeguards our body has in place. Excellent description of the multi-hit hypothesis. That is true. Oh, we should do I almost said we should do a cancer stream, but for some reason I'm hesitant. I'm hesitant to do a cancer stream. I'll talk a little bit about this just because it gives you an insight into how I think about things, because like it's not easy doing a biology channel, I think, because especially one that deals with disease quite regularly, because why do we care about biology, especially molecular biology? A lot of times it's for our human selfish reasons because we don't want to die. And I think cancer, it's interesting. I don't know if I would feel comfortable with devoting a whole stream to it, but it's something that I always think about. I just don't know how my audience would react if I were to, as like this goofy little cell, talk about. I mean, but everything I cover is tragic. This is why like I go back and forth. Like, am I being disrespectful or am I providing a service? It's really tough for me to say, especially when I appear in this form. Yeah. You could do an article thinking about how hallmarks of cancer. Yeah, I could. The question is if I should, because it is uncomfortable, I think. I mean, I don't know. I'll put up a poll because I don't know. In this day and age, like the fear of getting canceled is very prevalent in my mind. Knowledge is power. I mean, I do agree. But I'll see if my audience thinks it would be appropriate to cover something like cancer, just because I'm personally uncomfortable with... Well, the thing is, I'm streaming for you all. What matters is my streaming audience, because I specifically will never do a cancer video for my long-term videos, because that gets pushed by the algorithm. And who knows who ends up seeing that, right? I don't want... That's my fear, really, with the algorithm, is that if I do a formal cancer video, then somebody who has had a tragic loss is more likely going to see it and see... You know, because I do jokey things, but I'm never laughing at the disease. I'm never laughing at the suffering. I try to just keep the mood light, because not everything has to be doom and gloom. Yeah, it looks like we have the majority saying yes so far, although I'll leave that up for a bit longer just because I think it's tough. It's not an easy question to answer. It's a good idea because cancer has a lot of things people misunderstand. Yeah, it's also really hard is my other worry, is that due to the nature of my work, I do come across cancer biology semi-frequently, but it's tough, I think. It's, okay, I'll think about doing a cancer stream at some point, perhaps. As someone whose father passed of lung cancer, I appreciate when people are more lighthearted. That legitimately does help, because I've been fortunate enough to not, you know, at least yet, lost anybody in my own personal life. So I wouldn't know how I'd react to knowing that there's this goofy little white blood cell covering something as serious as cancer. But this has been helpful in terms of giving me permission, maybe, to talk about something like that. Not to say schizophrenia isn't serious too, but for some reason, cancer to me, I don't know, not to like rank horrible diseases or rank horrible conditions, but for me personally, that feels like there's a heavier gravitas. I suppose if you're watching me anyway, you don't mind that I'm a white blood cell talking about things like schizophrenia and disease and stuff like that. Okay. Thank you. I did a video on prion. I know, but my justification about prions is that the incidence is so rare that the like whatever, if you had to quantify the total human like a misery, I don't know. And again, I mentioned in like the comment that I pinned in my videos that like I call prion science cool. Like I'm very like careful because I'm aware that I'm not like a serious man talking into a camera, that these things are horrible. I just want to convey passion in understanding them, because I think you can be both. But these days, it's so scary, the idea of being canceled. If we're granting you permission to make videos, here are all the health conditions I grant you permission for. I don't think I'll ever cover autism. I'm sorry. I'm not nearly qualified enough. And it would be wrong as someone who is neurotypical of me to cover autism, just because I don't even know how badly I could screw that up. I am hyper and keenly aware that science has treated the science of autism has been not great in terms of communication specifically. And so I'm aware that maybe I should not be the one to communicate about autism, although if there's ever an opportunity for me to collaborate with a content creator that has autism who can moderate the way I talk about autism, that would actually, I would be okay with that. They would have to be very brave to moderate me, though, to tell me to do better, because I want to be sensitive for that kind of thing. Could I cover how common poisons work? I'm actually not a toxicologist, so I would be very amateur in that regard. Although there are toxins that I am intimately familiar with because I use them in my graduate research. Okay, but okay, that was a fun, quote unquote, fun little tangent. It sounds like a great way of bringing awareness for neurodivergence. Would it though? Okay, so here's the thing, and this is something for you to all chew on, because I think this is a really interesting discussion that warrants some talk. Okay, because as I am very hesitant to talk about cancer, and as I am very careful to moderate my image and to moderate to balance lightheartedness with the topic and the levity of real people with real issues who are actually suffering, what benefit would an audience get understanding the science of autism? That is my greatest hesitance, because ultimately, from my perspective, understanding the molecular biology behind something like autism doesn't necessarily make your life better. And it can also give people weird ideas about what autism is. Like, I just don't think it's a good idea this is something called biological essentialism, right? So biological essentialism is this philosophy that you are your body, that you are your biology. And I think the topic of neurodivergence is beyond biological essentialism. I don't want to reduce something like neurodivergence to molecules. I think that would be not moral for me to do, especially not without someone who has autism as someone who can moderate the way I talk about that kind of science. So that's why it's a little tricky. Does that make sense? You can do the cancer stream by talking about cell application instead. That will give you more leeway. I'm curious what you mean by that. I'm not entirely sure what that means. I can definitely appreciate your consideration of sensitivity, though, especially with cancer and autism. Yeah, these are not... It has a lot of potential for abuse. Because the prion video, I make light of it, but no one is going to take prions lightly. Like, everybody knows that, like, if you know what a prion is, you know that it's terrifying. I don't have, like, no one is going to be swayed by my silly video that prions aren't terrifying. And most of my video is saying that prions are terrifying, although they're not like a clear and present threat to humanity on a global scale. You know, I felt comfortable making that video because if you already know about prions, you already know that they're not to be messed with. But with cancer, you know, and the internet has done a terrible job of this, of just... you know, people being horrible to one another. That kind of thing has been muddied. A video on the usual pathogens, like Algeria, and I don't know those other ones. Yeah. I think most of my content will be pathogens, so no worry about that. No pressure making that type of content you're not comfortable doing. I'm open to the idea if and only if there's a content creator with autism that trusts me and I trust them. But it would be very tricky. Prions are just a couple of mutations away from being an existential threat. Although I suppose you can say for that any biological pathogen. The forces of natural selection I think are keeping that kind of apocalyptic future at bay. But let's not talk about apocalypse. Okay, let's get back to the stream. I think that was a very good and important tangent, especially talking about like the idea of this channel. But let's get back to talking about genetics, why don't we? Multicellular parasites. We'll talk about those at some point. So biology is a spectrum. I'll say it over and over again to prepare you for the big video that I do about biology being a spectrum. I don't know if I'll do the video, but I have a video in mind where I try to argue that you should not take biological categories very seriously, if you know what I mean. Autism is more complicated and different person to person. I think you made the right choice with that. Yeah, that's kind of my feeling. Okay. Not only are genetics complicated, but you wrap all of genetics into a package, and then you have to think about how the environment interacts with your genetics. And that's a huge headache, right? That's really, really tough. That is not an easy discipline. That is why I have a lot of respect, but a lot of wariness about the field of genetics, right? And Aiden might disagree, considering Aiden is a geneticist, but the environment and genetics, this is what makes this science so crunchy and complicated and tricky a lot of the times. And for me, as someone who likes thinking about molecules, this is what turned me off of genetics. But I recognize that this is an important part of the scientific process. And while I might not like doing this kind of science, I certainly think it's important to read and understand this kind of science. Wait, the environment changes your genetics? Not quite. It's more like your environment can affect the way that your genes are expressed. So, you might have a certain code in your DNA that determines how tall you will grow. But if you do not have sufficient nutrition, you might not ever get to that height, right? Stuff like that. The environment plays a huge role on how your genes gives rise to how your body functions. Aiden is saying that we're talking about epigenetics. That's also true, that there are factors that can affect directly your DNA, but that I'm not getting to for a while. Okay, so this is a big headache, right? This is a big headache. And again, I mentioned it before, but I like thinking about how molecules bump into one another and then they bend and then they do stuff. For me, the level of biomolecules is the fun area. I think geneticists and bioinformaticists are all nerds. And, if you're in science, you're a nerd, but like, if you're a geneticist or a biostatistician, I think you're like an uber nerd. And like, this kind of stuff is so complicated and there's so much math involved. But dork, yeah. But thankfully, I don't have to cover bioinformatics on my own because I am going to be collaborating with a fellow VTuber, Resta Makarabe, next week about bioinformatics. So if you are a programmer, I think you're really going to enjoy next week because Resta is going to take us through a case study in how to apply programming to solve biological problems. And like, I'm always really surprised at how much work my collaborators want to do, because again, if you're watching and you want to collaborate, totally cool. You don't have to do as much work as these collaborators have done. Like Clockwork and Resta and Nep and Sai, like they've all like worked really hard behind the scenes for collaborations on my channel. You don't have to do that, but Resta has or is preparing a whole presentation where we will walk through a programming problem, and I'm just gonna get to sit and chill and enjoy that with the rest of you, which is very fun. My brain desires to turn off right now, but I'm trying to keep it on. And Kuro, I mean, if this ends up being the stream that you fall asleep to, I am a fan of falling asleep and sleeping in general. I'm not a nerd. If you're doing genetics, especially if you're doing medicine, I don't know. I think that puts you in nerd territory. How extreme must an environmental factor be before it affects gene mutation? There are a class of environmental hazards that will cause gene mutation, UV being one of them, and there are also molecules that can do funny things to your DNA. Whether or not I talk about them eventually will do at some point. If I call me an Uber nerd, I will wear this badge with honor. Yeah, I think like math and programming, and physics, I think are the purest expression of being a nerd. It's just numbers. Wish I could have stopped by for longer. Deep and crunch time for final edits. Keep crushing it. Oh, I just mentioned you, Clockwork. Thanks for tuning in. Will it be an R? I actually don't know what rest of the program's in. I don't want to do it in R, though. I am a Python ride or die. I refuse to learn any other language. I had a physical reaction to R being mentioned. If we're lucky, we might get MATLAB too. I have a sticker that says she loves MATLAB. I love that sticker so much. It's so fun. Imagine that stream, there's no slides, just streaming an R session. Oh, that would make you too happy, I think, more juice. No, there will be slides. It's going to be story-based. I did tell Resta, and Resta is very cognizant of my presentation style. It will be a story format and not a coding session. R is pretty similar to Python, so it would be difficult for you to transfer over. I have very low frustration tolerance, and so I think doing any more programming that I have to do is frustrating. Nerd tier list. I would argue that chemistry and biology, for me personally, are about the same level of nerd. But yeah, I think math is just like truly the purest expression of nerdom. A stream, but only in PyMol. Stop. Okay, so this is a tease for next week's stream. We're going to be talking about programming. We're going to be talking about it with Resta. That's going to be super fun and exciting. To give you a little tease, it's going to be about how we can use programming to potentially fight bacteria. I think that's what the idea of the stream is. Just using programming to analyze bacteria and develop strategies to stop them from reproducing. Very, very exciting. Okay, so let's talk about schizophrenia now. So the way that I structure my streams is that I always want to give you the 101, but then I want to end on something very complicated, because I think the 101 stuff, you have to know it, right? You have to walk before you can run, but then I want to run. And where I want to run is talking about the very complicated genetics of schizophrenia. And so where I am going to be how I'm going to be doing this is I'm going to be talking a little bit about what schizophrenia is, and then we're going to be investigating what makes studying something like schizophrenia so difficult, but also so worth doing, as well as looking at some interesting and exciting recent findings about the inheritance pattern of schizophrenia. Okay, so schizophrenia, I don't even know if I should call this as a disease. Schizophrenia is a condition in which patients develop psychosis, leading to delusion and hallucinations. The diagnosis for schizophrenia is made by clinical evaluation. Okay, so this is why this is important, right? So for diseases like sickle cell, you can take it. Well, I shouldn't be talking about through my ass. Hypothetically, for something like sickle cell, you could get a blood test for that, and it would be a molecule that shows up, and you would be able to look at those blood cells underneath a microscope and get a biological output and say that's sickle cell, right? So those are diseases in which the underlying biomolecular mechanisms are known. Schizophrenia isn't like that. You diagnose schizophrenia, I mean doctors diagnose schizophrenia based on the symptoms that a patient comes in with. And that's because, as of right now, to my knowledge, there aren't really good biomarkers for the development of schizophrenia. But interestingly, even though there's not like a blood test for schizophrenia or, a genetic test for schizophrenia, schizophrenia runs in families, and there's about a 64 to 81% heritability rating between monozygotic twins, that one of them will develop schizophrenia. So monozygotic, mono meaning one, zygote meaning zygote. Monozygotic twins are twins that came from the same zygote, aka identical twins. So there is a 64 to 81% chance that if one identical twin has schizophrenia, the other will develop schizophrenia. But it's not 100%. If schizophrenia were entirely genetic, even if it's polygenetic, even if there are 100 schizophrenia genes, if you have identical twins, theoretically, those two twins should both have schizophrenia, but that's not true. In fact, schizophrenia only shows up 64 to 81% in identical twins. So that implies that not only are the genetics complicated, that there might be a large environmental role as well. So even though the heritability is quite high, I mean, at the high estimate, 81% chance one identical twin has schizophrenia, also the other twin has schizophrenia, that implies that the genetic linkage is decently strong. So then the question then becomes, why haven't we found the schizophrenia gene, right? If it's so heritable, have the scientists just been sitting on their butts, sitting on their hands, not doing the work? Even if it's multiple genes, that's not necessarily the magnitude in which this is a difficult disease to study goes beyond multiple genes. But you are sniffing up the right tree, Luke. That part of why schizophrenia is difficult to study at a biological level is that there's not just one schizophrenia gene. There are many risk alleles, and we'll get into what that means in a little bit. But that doesn't mean it's hopeless. I want to present this as like a David and Goliath type situation. Schizophrenia is one of these things that is so difficult to study, but the progress that has been made is very, very impressive. So biology is very complicated, right? I showed both of these figures before. So there's this central dogma, extended universe, and then there's this environment interaction with biology. It's really, really complicated. And both of these factors are going to be implemented in our study of schizophrenia. This is why I wish psychology is considered a science. My school doesn't categorize us at one. Ooh, Cideray would have a fit if she were to hear that that's how your school does that. Yeah, I actually don't know if schizophrenia fits in the category of psychology. It's definitely psychiatry. I don't really know too much about whether or not psychology, if that would fit under the branch of psychology. So biology is complicated. Schizophrenia is also complicated. The difference between psychology and psychiatry is psychology plus medicine. So schizophrenia is also complicated. So it's, biology is complicated, environment is also complicated. Schizophrenia as a condition is also complicated because, as Luke, you had correctly predicted, there are likely hundreds, if not thousands, of risk alleles. And the primary connection between neurobiology and the manifestation of the disease, or condition, I should call it, is not totally clear, and it's likely really complicated. So again, that's why you can't just take a brain scan and go, that's schizophrenia, right? Whereas other actually, I'm going to stop there because I don't want to propagate things that I'm not 100% sure of, but something like Alzheimer's can be diagnosed post-mortem because you can look at an Alzheimer's brain and point to the brain and be like, that looks like Alzheimer's. You can't really do that with schizophrenia, which is why its diagnosis is made by clinical evaluation, how the patient acts. So not only is this a polygenic condition, but also the brain itself is really, really complicated, right? The brain is so complicated. We know a lot about how the brain works, but we're not even close to figuring out how it works in its entirety. And the other thing that makes schizophrenia difficult to study is we can't exactly give a mouse schizophrenia. This is a major impediment in all research concerning neurological psychiatric diseases, especially psychiatric conditions, right? Because with certain neurology, there's a bit more of a deeper connection between us and a mouse, but with psychiatry, with diseases or conditions that affect the lived mental experience of an animal, it's hard to have that happen in a mouse. You would not know whether or not you were successful in making one of these mice. Avril Lavigne could have been a biologist, so complicated. True. I anytime I read about the brain, I should put on that song just to remind myself. I've seen headlines about a mouse with autism. How is that different? Are they just making it up? So this is a great example of how we're going to be able to talk about nuance. It's debatable whether or not you can give a mouse with autism. Because autism is such a uniquely human experience, because the way that we define neurodivergence is divergence from neurotypicality, right? Because we have this broad spectrum of lived human experiences, and then we kind of coalesce into two broad camps. But think about how autism manifests in a human being, and think about whether or not that can really happen in a mouse. You can have a mouse that is less social than another mouse, sure, but is that autism? Or is the mouse just not like being friendly? You know what I mean? Like, just because something looks like autism in a mouse doesn't necessarily mean that it is autism. But that brings up a good question. If that mouse doesn't have autism, is that mouse worth studying? I think the answer is still yes, because while the mouse might not have autism, it might have enough in common that it's still worth trying to understand. But again, that's why like biological essentialism is flawed, especially in the case of lived human experiences. You can't say that's an autistic mouse. You can say this mouse recapitulates factors of human autism. And so you always have to have that caveat, because it might not mean anything at the end of the day. Studying Alzheimer's and Parkinson's is made a lot easier because of markers, something that's really easy to take for granted. Yeah, absolutely. Although the mouse models have debatable usefulness, although I'm sure that people agree that those models are useful. How useful they are is a matter of debate. We managed to put Alzheimer's in a mouse. Again, I don't really... I've done quite a bit of reading about Alzheimer's, again, for my job, but also because I, at one point, wanted to study Alzheimer's. What we actually did is we crammed 6 copies of human Alzheimer's protein into a mouse and forced that mouse to produce Alzheimer's, where in reality, the manifestation of Alzheimer's in humans is a lot more complicated and age-dependent, right? Mice only live 2 years, 2.5 if they're lucky, and they live and die very quickly. We don't really know what role our slower aging process and our unique aging process has on the development of Alzheimer's. Also, there's all those things about gut-brain axis that's unique in humans. And yeah, again, the mouse recapitulates factors of Alzheimer's. We crammed the human Alzheimer's protein in their brain. But is it Alzheimer's? Why mice? Why not like raccoons or something? Mice are very easy to work with. They breed very quickly. They're very cheap to maintain. They are well behaved. It's not that hard to keep them relatively happy. Again, because even though we talk about animal science, we don't like want to do cruel experiments on animals, right? Mice, we know what a stressed mouse looks like. We know what a miserable mouse looks like. And we have the tools to give them a pretty okay life in their cage. And something like a raccoon, you're going to need like a jungle for, because they're too smart. We always say Alzheimer's model in C. elegans, for example. Exactly. Models. It's always important to keep in mind that they're not perfect. Probably cheaper to take care of a mouse than a raccoon, exactly. How similar are mice brains to humans? Depends on what you are asking. In terms of olfaction, they're really good at smelling, better than we are. Bad vision, so not great for eyes. If you want to study vision, that was historically done in cats. Basic motor stuff, right? Like they're connected the same way we're connected. If you want to study how mice move, and that can apply to humans, that's not too different. But for neuropsychiatric disorders, you have to be very careful with how much you want to believe in mouse models. So why do we say the mouse with autism model? But we don't say mouse with autism model. If they do, then that's being irresponsible. It's mice with traits that reflect autism in humans. That's like the full responsible thing to say, just so that people don't think that the mouse actually has autism. Mice being tame is debatable. That's true. There are certain breeds of mice that are more aggressive than others. Okay. good little tangent. Very fun. Good conversation. Let's get back to schizophrenia. Good transition back here. Okay, so biology is complicated, but we have the power to study it. So it can be kind of overwhelming to be like, oh my God, there are over 100 schizophrenia genes. How are we ever going to figure this out? And the thing that you learn as a scientist, or as you travel in your career as a scientist, is yes, problems are very complicated, but a good scientist knows how to break down a problem. So scientists, broadly are working on schizophrenia, but individual scientists are working on individual aspects of schizophrenia, and that's what's so encouraging about this. So you have people like Aiden, who are geneticists, who are up here working about the underlying genetics of schizophrenia to learn what biological factors go into schizophrenia. Then you have people like me who are at this level, cell biology and biochemistry, thinking about molecules, thinking about cells. And then you have the doctor scientists who think about pathophysiology. So how do molecules and cells interact at the organ level, we'll say. And then understanding this underlying biology, you have scientists who are developing and engineering therapeutics to try to address these biological problems. You also have social scientists who are working on and I don't actually know what you would call them. I don't actually know what you would call scientists who study the interaction between environment and disease. Epidemiologists, maybe. Someone might want to catch me on that. But you have scientists who study how environment interacts with genetics, or what environmental factors go into manifestation of disease. And then you have scientists who try to mitigate or try to figure out how to mitigate environmental factors so that you don't turn on schizophrenia, or however you want to say it. I found an article in Nature saying brain mapping across 16 autism mouse models. So 16 autism mouse models, that's the important part. So it's not mice with autism, because that means something different. That implies that they genuinely have the condition known as autism. But autism model acknowledges the fact that they are imperfect. It's just a model. My PhD advisor used to always say, all models are wrong, but all models are useful. This is something that was readily apparent in chemistry, right? We all learned that an atom is a collection of protons and neutrons in a nucleus, and there are rings of electrons that surround them. That's useful. It helps us understand a lot about chemistry, but it's not really right. It's a model. It's not correct. Then you learn about orbitals and up, down, and Pauli exclusion principle, and then all that gets to be a headache, and probability clouds, and then you realize those are models too. They're models that are more accurate than the Bohr model, and they're more useful in certain ways. So just because something is a model doesn't mean that it's necessarily reality. Yep, chemistry is like mostly probability. That's why I don't like chemistry. I'm sorry to all the chemists out there, but I felt it was very hard to feel grounded. Whereas in biology, I'm like, that's a cell. I can see it with my eyes. I don't have to think abstractly. I don't have to do math. I like things that I can imagine. Ah, yes, the electron cloud and resonance. My brain just went poof. Yeah, sorry for bringing back that chemistry trauma for all the biologists out there. So, if you are a geneticist and you want to study something as complicated as schizophrenia, there is a very powerful yet flawed tool called the GWAS. And if Aiden is still in chat, maybe Aiden can catch me on this, because I am not a geneticist, and here is the part where I am mostly going off of what I learned in graduate school. So a GWAS is a genome-wide association study. So you're looking at all of the gene variants in a human, and it aims to map out all the variants in a gene associated with inheritance of a disease. So you go in and you don't really have a hypothesis. You just say, you get a population of people with schizophrenia and people without, and you say which gene variants are the most associated with inheritance of schizophrenia. And then you try to figure out if that has any meaning. I've run these before. Perfect, Aiden. So you can call me if I'm being too liberal with my description or need a little more nuance. These variants are called single nucleotide polymorphisms. So this is a very fancy way of saying places in the gene where the letter has been swapped. So your genetic code is made up of a long list of letters A, G, C, and T, and all sorts of different arrangements and combinations. And a single nucleotide polymorphism is one individual location in the genome where you have a potentially different letter at that exact spot than the rest of the population, or most of the rest of the population. So these SNPs, these places where we each individually differ, you can almost think of them as your genetic footprint or fingerprint, right? Because your combination of SNPs, the combination of little tiny one base pair variants, that's what makes you you, right? That's uniquely you, your genetic fingerprint. The thing about GWAS studies is that they are very powerful. You can get a lot of data, but they are subject, and again, Aiden, correct me if I'm wrong, they're subject to bad data, bad design, and bad analysis. So I'm not big brain enough. I'm too in the middle of the nerd spectrum to be able to tell you how GWASs are flawed. But a lot of the time they are, especially when you deal with big data sets or data sets that aren't big enough. Statisticians can bicker with each other all the time talking about which test is statistically appropriate. I'm not smart enough to give you that journal club, but just take in mind that like whenever you see a GWAS study, you kind of have to take it with a grain of salt. And that's why I like my field. As Morju said, I can tell with 99% certainty that that is a cell. That's the kind of concrete science that I like. I'm a fan of being like, that's a protein, that's a cell. We can do this, this, and that. And then that's pretty solid biology. That is true. But yeah. So what did we learn in these GWAS studies? Well, when you do these GWAS studies on schizophrenia, you get a list of risk alleles. So these SNPs are little variants, and they are more common in people with schizophrenia than people without. And so that's not what I wanted to do in terms of animation, but we'll go with it. So what do we know about the risk alleles of schizophrenia? So we have things like glutamate receptors. So if you've attended my previous streams, you would know that glutamate, I think this was in the trivia stream, glutamate is the principal neurotransmitter in the brain, excitatory neurotransmitter. Glutamate is the way that your neurons talk to each other. Sure, there's dopamine, there's serotonin, there's epinephrine. Does epinephrine count? Gosh. What am I thinking? Dopamine, serotonin, GABA, acetylcholine from muscles. You know, those are all neurotransmitters, but actually the most important, most common one is glutamate because it's just the basic way your neurons talk to each other. And so some of the risk alleles are these receptors, and so that makes sense to me. Schizophrenia is a brain thing, and glutamate is a very important brain thing, so it kind of makes sense that a brain thing is involved in a brain thing, right? Yeah, you are spot on. You can't reliably draw conclusions based on GWAS alone. It's ultra important to keep in mind that correlation doesn't equal causation, something that I might sell as merch one day. Because if there's a statement that scientists should live and die by, its correlation does not equal causation. And that's, again, why I like my field of study, because we can actually do causation. Did I draw that neuron? No, Toxo did. Toxo, my mod, my partner, my artist behind the scenes. The rigor of this Phi the neutrophil. Do a popcat react. Yeah. So, yeah, I like my field of science because we can actually do causation, and that's very satisfying to me. But again, I don't want to poo-poo the work that geneticists do. It's just, it's not for me. It's not my taste. So it makes sense that glutamate is involved in schizophrenia because glutamate is involved in everything in the brain. It just makes sense to me. So what else is involved? Synaptic scaffolding proteins. So the area between two neurons where they communicate, that's called the synapse. So when one neuron fires to another, that space is called a synapse. And that synapse has to be built right underneath that little, and so if I can get my cursor to show up, yep. This is called the postsynaptic end, and this is the presynaptic end. And you can imagine that there's another neuron at this end. This is where this neuron would connect to the next neuron. So again, schizophrenia is a brain thing. I'm not surprised that there are risk alleles involved with the structure of the synapse. Yeah, Toxo is mama and papa. That is true. Not only did she draw Phi, she also rigged Phi. They clearly must be talented artists for being able to draw things that are both cute, have expressions, and are accurate, or at least look accurate enough. Yeah, don't tell the real immunologist who want me to have a trilobular nucleus. It's just not cute. Having Phi have a trilobular nucleus is not cute. That's where we're at with that. But I agree. I think all of Toxo's art is a big contributor to why the channel is as nice as it is, or as successful as it is. Like, that's also why all of your super chats go to buying Toxo a very nice sushi dinner. No pressure if you can't afford to, but I'm just saying, like, I've been keeping track of all the super chat money, and when we hit the omakase mark, we're gonna celebrate. Bless Toxo for being mama and papa. True. Yeah. So synaptic scaffolding proteins. I'm not surprised that schizophrenia, there's risk alleles involving how neurons talk to each other. That's not surprising. Voltage-gated calcium channels. Again, calcium is a major ion in the brain that helps your neurons communicate. Also not surprising. And then we have the dopamine receptor. And I'm going to take a break to read chat for a sec. We have to sacrifice scientific realism for the pursuit of cuteness. That's legitimately what Toxo's attitude was, and I think it was the right idea. You can have 2 nuclei for the eyes and 1 for the mouths. Yeah, but I think simple is best with our philosophy here. Or rather, I shouldn't take credit, because the only thing I contributed to Phi's design was, wouldn't it be cute if the nuclei was like an eyeball? That was my contribution. What does a dopamine receptor mean? Dopamine is one of the neurotransmitters in the brain, and it is commonly associated with the signaling for movement. So whenever you move, there's a ping of dopamine that helps your neurons communicate that you should move, but it's also involved in learning and reward. So it's like one of the 2 happy molecules, you could say, and is involved in addiction as well. Love your way of explaining. Thank you. Welcome to the stream, Prey. So I highlighted dopamine receptor here. I heard once that ADHD people have a deficiency in dopamine. I am not a psychiatrist, so I can't comment on that. I wouldn't be surprised, though. Be interesting to look at. That's a root word. When boomers say something is dope, I still say things are dope. You're causing me to apoptose. Sorry, getting off track. Why did I highlight dopamine receptor? So this is really, really interesting. Really, really interesting. And I think this is a really great opportunity to practice some critical thinking, some really tough scientific thinking. So the primary treatment for schizophrenia, and you can treat schizophrenia. This is something that is like very hopeful, right? Because schizophrenia, it's portrayed in the media like this horrible disease that makes you crazy. But what they don't talk about, and what's important to remind people, is that treatment for schizophrenia exists. Now, of course, the severity of schizophrenia can vary, but just because you have schizophrenia does not mean that your life is ruined. Although it can be quite severe. There are treatments that exist, and the treatment that exists, the one that I know of, is one that targets the dopamine receptor. Right? The dopamine receptor, so it's a medicine that blocks the dopamine receptor. So it lowers the amount of dopamine that is signaling, or that can signal in the brain, and that helps with the psychotic symptoms. Does that mean that dopamine signaling is what is wrong in schizophrenia? Knowing nothing else. This is a really interesting question from a scientific thought point of view. So I'm going to throw up a poll. We don't know a lot about how schizophrenia works at a biological level, but we do know, and someone in chat can correct me if I'm wrong, but we do know that the class of molecules that is the most effective in treating schizophrenia are dopamine receptor antagonists. So molecules that block dopamine from being used. Does that mean that schizophrenia is definitely a disease of the dopamine receptor? And so I'll put that as a poll. Knowing nothing else. And I'll put that as a poll. And longtime stream viewers might know that when I break out this tone, that it's a bit of a tricky question to think about. But it's not necessarily yes versus no that I'm interested in. I'm interested. Wait, am I allowed to say anything about schizophrenia, or will it be spoilers? We have some pretty high. We have some pretty high agreement in the poll right now. So, Kuro, let's hear what you have to say about schizophrenia. And then, well, actually, hold on with that thought, Kuro. I want to unpack why this is a interesting question, and some of you have already touched upon this. Faulty dopamine signaling could be another downstream condition of underlying issue or multiple. Yes, I think also General Mustache, you also had the same idea. So dopamine could be a part of the story, but it doesn't necessarily have to be the only thing that's wrong, or even the principal thing that's wrong. So something else, as Morjuice had said, something before dopamine might be going wrong, and then you fix dopamine later, and then that helps, right? So there are pathways in biology, right? Order and information that flows. And maybe schizophrenia maybe dopamine is like the last thing that is messed up, and then you treat the dopamine, and then that helps schizophrenia. But if we're thinking about how to make a better treatment for schizophrenia, and if we're thinking about what the root causes of schizophrenia could potentially be, you're right. It might not be 100% dopamine's fault. You might just happen to be able to fix it by treating dopamine. Hello, the Clockhead. Welcome to the stream. Yeah, I saw you were in pre-chat. Not only that, think about another. Again, knowing nothing else. So dopamine could be downstream of what's actually wrong. What's another hypothesis? So here are the facts. Using a dopamine antagonist helps with schizophrenia. But maybe it's not the thing that is wrong with schizophrenia. What's another idea behind this? In software engineering, we do root cause analysis for software bugs to make sure we're fixing the actual issue and not just the symptom. I love that. I think that is a perfect analogy in how scientists have to unpack causality here. So this is a challenging idea, right? This alternative could be really hard to come up with. Could affect something else. Dopamine receptor antagonist could affect something else. Yes, that's a great point. So maybe, and this is certainly true for a certain, I believe, again, correct me if I'm wrong, Chad, if there's anybody who is more aware about psychiatry than I am, but there are dopamine receptor antagonists that also do other things. So just because we use a class of molecules that affects dopamine doesn't necessarily mean that dopamine is the thing that is being treated. That's a really good alternate hypothesis. Again, knowing nothing else. Again, this isn't, I'm not saying that this is true. I'm saying that to challenge you to think divergently, to challenge you to think scientifically, to challenge you in becoming future people who are going to shake up our understanding of science. These hard questions that try to break the veil or try to break convention, that is what we are going to need. People who think outside the box, and this kind of out of the box thinking is important. Absolutely. There's one more alternate. I think maybe there are two symptoms with a single cause. I'm not entirely sure what you mean by that. There's one more potential model, and is that I think maybe this is what Alexandros are getting at. Maybe you inhibit the dopamine receptor like the drug tells you should, but maybe that is compensating for something else going wrong, right? So maybe your dopamine is actually fine, and maybe when you use this drug, it's not fixing dopamine, but it's bringing dopamine down because something else is going wrong, and bringing dopamine down mitigates the effect. Maybe your serotonin is going crazy, right? Serotonin is the happy molecule, just as an example. I don't know enough about the wiring of the brain to make sure that this makes sense, but as an example, maybe schizophrenia is actually a disease of crazy serotonin happening in the brain instead. Maybe because serotonin and dopamine act similarly in certain locations, that bringing down dopamine is compensating for serotonin being too high. So you have 3 different ways that schizophrenia could be working biologically that all get treated the same way. And that is one potential reason why schizophrenia is so complicated, because there are so many genes involved, so many different ways that things could be screwed up, but it seems that they converge on this dopamine receptor. So that is interesting to me. So that's great examples that you have all said before. And now, Kuro, I will hear what you have to say about schizophrenia. Yeah. So, oh, by the way, for those of you who don't know, Kuro is a VTuber who streams on Twitch. I don't know if Kuro, if you want to drop your link in the chat box while you talk about schizophrenia. But I'm a big fan of whenever Kuro streams Minecraft. It's very relaxing and fun to be in those streams. I don't know too much about serotonin syndrome, so I would not be able to say. But yeah, so I hope that kind of helps to illustrate how complicated genetic circuits can be, or even science, really, because you would think, and again, for a long time, I thought dopamine, oh, that has to be the thing that's wrong with schizophrenia, because when you use a dopamine antagonist, people get better. But actually, in doing research for this particular stream, I realized that there are a lot of different ideas of how schizophrenia functions, and they're not necessarily mutually exclusive. They could all be somewhat true because schizophrenia is such a difficult to understand condition. Dopamine antagonists may be stopping dopamine from affecting the part that is ill. That is true. That is sort of the mainline hypothesis for why dopamine receptor agonist. And again, that's the most likely situation. I'm not saying that they're wrong to think that schizophrenia is a condition of the dopamine circuitry, but I'm saying that to, dot your I's and cross your T's, we should be critical and really thinking deeply at every step when we're thinking of new ideas, right? Because that's what we need, new ideas. Okay, I don't know, Kuro, if you're still around, but I'm going to head back, and if you're still around later, then we can talk about schizophrenia. Oh, there we go. So schizophrenia has both positive and negative symptoms. Positive things are like hallucinations and delusions, while negative are the lack of movement, becoming mute. Only positive symptoms are treatable because there's no treatment for the negative symptoms so far. Oh, that is interesting. I didn't know that lack of movement was a... Oh, that's so complicated, because dopamine is also a neurotransmitter that helps you move. So if schizophrenia is a condition which there's too much dopamine around, that could imply that the area in which dopamine is active is very important as well. Very, very interesting. Yeah. So even though we have a class of molecules that can help people with schizophrenia, this is one of those frontiers where, this next generation of scientists, current scientists in the field, I don't want to dismiss your hard work as well, but in terms of like looking forward to a disease that is going to be very hard to crack, I think schizophrenia, I'm going to be waiting on bated breath, because not only do I think just like intellectually is an interesting problem, but also it's like, again, never want to separate the condition from the human suffering. This is something that I have a lot of personal existential sympathy for, right? It's tough to live with this, and the amount of human suffering that we could alleviate by figuring this out, it would be amazing. Young people like you, yeah, exactly, young people like you, make sure you work hard in school and don't burn out. Okay, that was a fun little departure to think critically for a second here. Second generation antipsychotics are somewhat effective for negative symptoms, but not completely from my understanding. That's interesting. Toxo, do you know what the mechanism for second gen antipsychotics are off the top of your head? Because I'm only aware of anti-dopamine stuff. Late to the stream, was there any discussion on those who are born sightless and not developing schizophrenia? That is interesting. I've never heard of that. That is very, very interesting. Maybe for a future stream. Really difficult to tackle. Felt like if I mentioned the negative symptoms, it would have spoiled the answer. Yeah, I'm not a schizophrenia expert in the condition, but my graduate research overlapped with a particular aspect of schizophrenia that I can't wait to get to. This is such a fun conversation. I'm so glad that you're all here to hear me nerd out about something that I've been thinking a lot about, especially recently. Serotonin, I believe, is in second gen. Ooh, that's fun. Again, schizophrenia, not fun, but the idea that it is also serotonin. Okay, that gets into one of the slides that I'm going to get to. Okay, so we took a break to talk about the dopamine receptor. Other risk alleles include signal transduction elements. So for those of you who don't know, signal transduction, basically it's a way of amplifying signals that your cells get. So if you think about, your cells have to react from very quiet signals all the time, right? If your cell touches a protein, they have to somehow propagate that signal to the nucleus and respond accordingly. So signal transduction is a set of proteins and genes that help turn a very quiet signal to a loud signal so that it gets to the nucleus, so that the cell can actually react. So these are all like very general neuron and biology problems, which is frustrating because we would really like a very targeted risk allele that we could actually get our hands on. Cytoskeleton receptor and trafficking proteins. So the cytoskeleton, so the backbone of the nucleus, if you will, that's really important because that's the tract along the neuron's body is amongst the more important parts of the neuron because it helps you get proteins from one end of the neuron to the other, including stuff that you need to send signals. So that's important. You mentioned my stream, but I don't really do educational content, so probably not a common audience. I mean, I plug any of my mutuals who come on. No need to be shy about that. And synaptic plasticity proteins. So synaptic plasticity, this is something that I've spent really long in graduate education studying. So synaptic plasticity is the ability for the synapses, the space by which the neurons can communicate with one another, that area. In order to learn and to grow and develop, your neurons need to strengthen their connections and weaken their connections. You know, when you are born, you have like a zillion, gajillion neuronal connections. And then as you get older, you lose some of those connections. Does that mean you are dumber over time? No. Over time, your brain will figure out which connections need to stay and which connections need to go so that your brain is more efficient. And so this remodeling process, this ability to change, that's also implicated in the inheritance of schizophrenia. Okay, here is, and this is something where if you guess this correctly, I will gift one membership. I made a mistake by promising too many last time, but I will gift exactly one membership here. Who in the chat, and if you're a biologist and you know the answer, don't respond, especially if you're a psychiatrist. People who are born sightless can develop schizophrenia. I don't know what this is about. Sorry, it's just something I remember reading off a while back. Yeah, maybe there's some kind of connection, but you may be misremembering it, or I've just never heard of it. Okay, so chat. This is really exciting. This is really, really exciting. What is the region of the genome that is the most associated with schizophrenia? Any guesses? And this is in line with like my prion video where if you watch the 5-bit, you know, I asked, I talked about Doppel in the prion video, but I didn't say what Doppel actually did. But Doppel, you know, prions, which are in the brain, Doppel's in the sperm. And this is the same level of surprise that I had here because the most associated region of the genome associated with schizophrenia is none of these things. And that's really, really interesting. So, sexual reproduction? No, not quite. What is the most associated with schizophrenia? Just as a guess. Just guess. Take a guess. The end of the chain? Not quite a precise answer. I'm looking for like genes or bodily functions or organ systems or tissues or stuff like that. It's not related to Doppel. The answer to this question is not related to Doppel. But if you had to guess, what would you guess? Something with senses? Yeah. Okay, now if you're a biologist and you happen to know the answer, if you want to chime in and take a crack at it, that would be very interesting. Brain connections. So there's already synapse stuff on this list, so that's already been included, right? Synaptic scaffolding and synaptic plasticity. Ventricles, so Kuro's hypothesis is on circulation. That's a certainly reasonable sort of guess. The dopamine receptor, that would be the obvious choice, considering we talked about the dopamine receptor, but this is actually a not obvious choice. All right. A has got it, and I don't remember. I think A actually has a background in biology, or maybe attended one of the streams, because immunity, all of these things, the numbers 1 through 7, all of those things make sense for schizophrenia as risk alleles, right? Because those have to do with the brain. They have to do with how neurons talk to each other, and we know that schizophrenia is a condition of the lived experience. But number 8, across all of these GWAS studies, these genetic association studies, the most common feature in people with schizophrenia is located in a region of the gene that codes for immunity proteins. I will gift 1 sub if someone gets it right. A, you did get it right, but now be honest here. Did you attend my collab with Dr. Wendell or Clockwork Show? Because I teased this answer if you attended those. If you legitimately didn't show up to those, I'll gift a sub. But if you did, yeah, it's a little cheating. Immunity is the most associated region of the genome that has been related to schizophrenia. So what is this particular genome? This is the region that contains the genes of the major histocompatibility complex, MHC. Moving the goalpost, my streamer is a scammer. Until you make a scammer emote, I'll be fine with that. There's certainly no way that I would tolerate more members joining, so we have more spots for emotes so that you can make a scam emote. No way. Joke's on you, I barely remember specific things from streams I watched yesterday. That's fair. All right, so the major histocompatibility complex region of the genome. MHC is the protein that is near and dear to my heart. I think it's one of the most interesting proteins in existence. And what this protein does is it lies on the surface of cells, all your nucleated cells. And what it does is it tells the T cell whether or not your cell is infected. This is immunity. So A's first answer, immunity. It's not related to memory. So this cell, right, this pink cell, these are epithelial cells that Toxo drew. They're making a protein that informs the T cell whether or not they're infected. And the region that has this gene is the region of the gene that is most associated with schizophrenia. And that's crazy. Now, this region, and the reason why I'm so careful with this language, is that this region of the gene has other things that aren't MHC. It's not just MHC in here, but it's mostly MHC stuff and associated other genes. But this is the region where this is the most common genetic association. So I'm going to be taking from this open access review to talk about the connection of this region of the gene and schizophrenia. So this is my warning, again, as a scientist and as one that is trying to model good, rigorous, and unbiased thinking. Immunity stuff is my bias, obviously, right? Like, I am a neutrophil. I love immunology, even though I never studied it in a very, very formal context. My graduate research overlapped with immunology by quite a bit. And so I am partial to accepting that schizophrenia has a major immunological component because that is my bias, right? Your bias might be insert K-pop joke here, but my bias is immunology, right? Is MHC a complex of proteins? Technically, yes. It's made out of the heavy chain, and then it has a part called beta-2M and beta-2M that is on the cytoplasmic side that helps. But it's, I don't actually know why they call it that, because plenty of proteins have 2 subunits or multiple subunits. Yeah, I never learned actually why it has that name, which is kind of interesting. I think I read this in a book somewhere. Okay, it's a little, maybe I'll gift, maybe I won't. We'll see at the end how I feel. So immunological mechanisms are my bias, but there are plenty of other scientists that have different ideas about how schizophrenia functions. And that includes dopamine dysregulation, which is, I think, pretty common, like pretty popular. Glutamate dysregulation, serotonin dysregulation. So this one is fun because of what Toxo said, that second generation treatment for schizophrenia that has a serotonin component can help some of these other symptoms. So maybe it's a combination of dopamine for the hallucinations, serotonin for the movement stuff. So again, these are not mutually exclusive. Just because it has an immunological component doesn't mean that it's all of schizophrenia or even most of schizophrenia. It's just the most shared among schizophrenia. Possibly a silly question, but what immune system activity is seen in the brain? Oh, thank you for asking this question, Alexandros. I spent 6 years in graduate school studying this very specific question. So, some of you may have seen this rant before, but it's very interesting to know that when I grew up, we thought the brain was immune privileged because there was the blood-brain barrier, right? You don't want neutrophils and T cells hanging out in the brain because the work that we do is very destructive. And some regions, like your gut, can afford to lose a few cells. Your neurons cannot be afforded to lose. You can't afford to lose your neurons, basically. Is there neuronal regeneration in the brain? Sure, but not that much, right? Not that much. And so it was thought for a long time that the immune system and the brain did not interact, so the brain was called immune privileged. We actually know now that that's not true. The brain is filled with immune proteins doing all sorts of different things. What's true is that the blood-brain barrier is still intact. If you have T cells in your brain, you're in trouble, right? If you have a traumatic head injury and you get infiltration by neutrophils, you're in trouble. So the brain is very much privileged from contact with the immune cells of the body, except the microglia. The microglia are immune cells, but they're the brain's immune cells. They're specialized to operate in the brain. But immune proteins actually have a function in the brain that they don't have anywhere else, and that's crazy. So I talked about synaptic remodeling earlier. Over time, as you learn, so learning has two parts. When you learn, your neurons strengthen connections that need to be strengthened and weaken connections that need to be weakened. And combining those two is what makes your neurons be able to learn. And the proteins that help your neurons trim the connections that they don't need are the same proteins that help your cells blow up bacteria. Absolutely crazy. I love the overlap of the immune system and the nervous system. And they're also evolutionarily related. So the nervous system and the immune system, they're actually like cousins in a kind of way. And that's really, really fascinating. I remember the brain has specific macrophages. Yeah, those are microglia, the resident phagocytes of the brain. So I love talking about immune and brain stuff. And it's also why I wanted to talk about schizophrenia, because this has a particular immunological overlap with my research interests. So not only is serotonin, and again, thank you to Toxo for contributing that information, with serotonin being a part of the next generation of psychiatric medications with schizophrenia, but you also have GABA. So GABA is the depressant neurotransmitter. It's the neurotransmitter that quiets neurons. And there's also a not completely, but like a parallel school of thought that suggests that schizophrenia is a neurodevelopmental disorder. So not a disorder of like signaling, but like your brain developed in a kind of way where it has lent itself to schizophrenia. And again, these aren't mutually exclusive, but they're different like schools of thought. Brains saying they love brain stuff, kind of base, not gonna lie. True. Why would you want them to be quieted? The best analogy that I can think of is why we did not evolve photographic memory. So if you are studying to be in medical school, you would imagine, oh, it'd be really helpful if I was just able to remember everything in my entire life. But is that really true? Because something that's really helpful for just basic happiness is being able to forget when bad things happen to you, or to, if you have an embarrassing interaction, like if you're at a restaurant and your waiter says thank you and you say you too, like that is an interaction you would prefer to forget, right? So that's an example of why losing interaction is helpful, because it would be very encumbering to have your brain on the entire time. So it's more about fine-tuning certain interactions to make them more efficient. Your brain is incredibly cost heavy. It chews up glucose like no tomorrow. You want your brain to be chewing up glucose in an efficient manner, not connecting neurons that don't need to be connected in terms of plasticity. And in terms of GABA helps with the balance between excitation and inhibition, because not only do you want a good, not only do you want the neurons that you want to be not only do you want the neurons that are doing what you need them to be doing that thing, you also want neurons that are not supposed to be on to be off, is how I like to think about it. Is someone cooking? I'm always cooking. Oh, you mean the sound in the background? We haven't come up with a name for the cat yet, but the cat is getting fed. I don't know if you can hear her meow. Yep. Can't call the cat Toxo because Toxo is Toxo, so we haven't come up with a good name for her yet. Serotonin syndrome is terrible. My doctor messed up some dosages and I spent the day in the hospital. Seems bad. Yeah. A lot of, that's why I'm personally invested. Like, you know, I cover a lot of pathogens and stuff too, but my other interest is a lot of these neuropsychiatric, neurological conditions, just because, again, they take away a lot of, you know, potentially taking away a lot of human experience. And I think that is a worthy area to specialize in, personally. Is calling a cat meow valid? It's very valid, I would think. Maybe we'll have a poll, I don't know, at the end of the, a name for the cat, natural killer cell. I like that. At the end of the stream, we'll come up with a name for the cat, so I can refer to her as something other than just the cat. Okay, so yeah, there are different schools of thought when it comes to understanding what schizophrenia is. Immune is not the only one, nor is it mutually exclusive. So, to me, this makes a lot of sense. I don't know if you should let chat name your cat. I'm ultimately going to be the decider. I'm not going to fall for the Bodie McBoatface trap, but chat can suggest, and then I'll pick from the suggestions. So it's not a surprise that there's some immunological component to schizophrenia, because schizophrenia does have clinical attributes associated with immunology. Cat Sully popped us a trigger. Save this for the end of stream. I really love some of these ideas, and it does, because I have been agonizing over a good name and I couldn't think of one. So we know that schizophrenia statistically is associated with prenatal infection. Now, does that mean that prenatal infection causes schizophrenia? And again, Chad, I'm asking this question to you. Does this mean that prenatal infection causes schizophrenia? Why or why not? Excuse me. First question, prenatal infection. So we know that when mom is sick during pregnancy, that there is a higher chance that the child gets schizophrenia. Does that mean that mom being sick during pregnancy caused schizophrenia? We'll give a second for the votes to come in. Correlation, not causation. Correlation, not causation. Yes, my goal for my streaming career is eventually that 100% of my chat will be able to tease that apart. But I'm very happy with where the poll stands right here. Correlation is not causation. Okay, so if we take the assumption that it's not actually the prenatal infection that's causing schizophrenia, what else could be correlated with prenatal infection? We're being conditioned, exactly. That's my... The thing is that if I were to write a curriculum for high school teachers, and I'm talking out of my rear because I don't think I would be a good high school teacher, but my dream curriculum for high school would to include discussions about correlation and causation, because I didn't get that as a high schooler. And I think to help people just like function in our modern information era, understanding this difference is so important. Bad standard of care, congenital conditions. I think bad standard of care is more where I'm heading towards. Anytime 5 pushes at a pull, it always means the obvious answer isn't correct. Yeah. That's one of those teaching meta secrets, that if you ask a leading question in this way, you give the students to participate, you let them challenge a challenging idea, but then most of them will get it correct and then they'll feel good about themselves. It's my 5,000 IQ play. Yeah, so many conspiracy theories are started by not quite understanding correlation and causation. So bad standard of care, what could that be correlated with? A big thing that crops up over and over again in terms of correlation and health outcome. If prenatal infection is correlated with schizophrenia, what might prenatal infection be correlated with? What group of people is the most likely to not get sick? Most likely to not get sick. Prenatal infection may be causing something to be born early, which often comes with some neurological problem. That's an interesting hypothesis. So maybe not the infection, but stress to the mother in general. That's definitely an idea. Rich people, yes, Luke. Polymerase. Oh, that's such a good name too. Oh, well, let's revisit all of them at the end. Yes, rich people. So something that can be associated with prenatal infection is standard of living, or just raw money, right? So it could be the infection, but it could also be money. I don't really know if they've done those studies, but again, when you think about associations, you always have to be very, very careful. Doesn't necessarily mean that infection equals schizophrenia. So if you know a pregnant person and they get sick, you don't have to freak out. Oh, baby's gonna have schizophrenia. Don't freak out. It's a numerical association. It's not causation. Okay, but it is statistically in the conversation. Polymerase. Oh, that's pretty funny too. Polymerase definitely rolls off the tongue a bit better. Autoimmunity. So people with schizophrenia also have a higher chance of having autoimmune disorders. So actually, I think in the Discord, I think it was I Caused a Huge Note, wasn't exactly sure, but that someone had brought up the idea that schizophrenia is like autoimmune disorders. And here's where I would say that that's true, in the sense that they're both genetically very complicated, and also they happen to associate with one another. Increased inflammatory markers, so people with schizophrenia also have increased inflammation. Again, correlation and causation. We don't know if the increased inflammation is causing schizophrenia or if they have the same root cause, right? Always important to keep that in mind. Maternal immune challenge in mice. So if you take a pregnant mouse and you expose that mouse to bacterial protein, it will cause mom's immune system to freak out. And that process is associated with schizophrenia-like symptoms in mice. And again, there are a lot of problems with this. It doesn't necessarily apply to humans, so don't freak out. It's not worth freaking out, okay? Take a deep breath, especially if you're a hypochondriac, right? It's a study done in mice. Mice are not humans. Also, mice can't really get schizophrenia as we understand it. But having mom activate her immune system in a mouse has some statistical association with the development of schizophrenia. So that's interesting to note. And then, independent of these ideas, is the fact that genetically the MHC locus is the most common factor across all people that have schizophrenia. How does autoimmunity not instantly kill you? I mean, it kind of depends. Location, severity, etc., etc. You can have varying degrees of severity with autoimmunity. And again, biology is a spectrum. Not everything is on or off at the same time. Everything is a gradient. Everything is fuzzy. Let me say that louder. Biology is a spectrum. One imagines testing mice for hallucinations might be a bit difficult. Exactly. So take these with a pile of salt. It's not worth freaking out over a study done in mice, but it is a contributor for our understanding of biology. Okay, so it's not surprising then that there's immunity as a potential contributor for the development of schizophrenia. So why haven't we nailed down the genetics yet? So like, we know that immunity is associated, but we haven't established a causative relationship. We don't know whether or not immunity causes schizophrenia. So what the heck is the holdup, right? Like, why is this so hard? Number one, correlation is not causation. Exactly. Yes, I should make all of these a shirt. Correlation is not causation. Biology is a spectrum. My mantra is to live and die by. Rules of Fight Club. Chat, you are on fire today. Chat is going so hard right now. Okay, so schizophrenia is a difficult problem. We know that there is an immune component. So if we look at the MHC region, what stuff is in there that is associated with schizophrenia? So now we're looking at a gene by gene level, not just the MHC region, but individual genes. One more spectrum I'm in, the biological spectrum. That's really funny. Yeah, chat is killing it today. So when we look at the MHC region, we can look at individual genes that are associated highly with schizophrenia. So we have RPP21. This is a tRNA processing gene. To me, I'm sleeping on this one. tRNA processing, this is so vague. This is so important for the entire rest of your body. It doesn't scream to me as something brain specific. I wouldn't even know where to go with that. Like, I don't know I wouldn't know where to start with trying to fix that. Notch4, which is a neurodevelopmental gene. Now we're talking more in the realm of neurobiology, in which case this, to me, is a more tempting target than RPP21. It's impossible that all those are involved somehow. Yeah, it is true. It is true. But in terms of which target I would pick, tRNA processing is so vague and central to cellular biology that, like, if your tRNA processing is a little screwed up, but you're alive and you try screwing with it again, I don't know. For me, I wouldn't go after that one. Then we have something very, very interesting, and it's proof that biology is evolving. And I mean, what I mean by that is that when I learned about biology in high school, I was amazed at how much we actually knew about the body already. Like, I seriously had a conversation with myself being like, do I want to be a biologist? It feels like we kind of figured everything out already. I mean, we know how genes are inherited. We know how all of these molecules work. How much left is there? We're talking in the year 2024. There is a major gene associated with schizophrenia for TRIM26. We have no idea what this protein means. Zero idea. Zero idea what this protein does. One of you in the audience might be the one to crack that code, right? I was very haughty as a kid. I have a big ego, which is why I was like, oh, well, it looks like we're done with biology, I guess. But yeah, in the year 2024, still a major protein in the brain. Actually, I don't even know if TRIM26 is in the brain. It's a protein of completely unknown function. Have we tried sticking in a mouse yet? You could be the one to do that. I don't know. Not as far as I have learned. We don't know what TRIM26 does. Icosahedron. Maybe infection early has to do something with MHC region allele. Yes, I think that is definitely a possible mechanism of action. That MHC is mediating some kind of immunities. Maybe it's doing it in a weird way, and that's causing issues in schizophrenia. Sort of a more advanced thing to consider, but things like ADAR editing of mRNA also really complicated things. Oh, in terms of genetics, yeah, I can imagine. You know, one thing I learned from science is that no, it's never the case that a field is completely figured out. I just wasn't creative enough in high school. And then we have chromatin remodeling genes. This, like RPP21, I just don't know what to make of this because chromatin remodeling, it's just so vague and not really specific to the brain. I wouldn't pick that as a target personally. I would be really interested in what TRIM26 does and how Notch4 can potentially contribute. And of course, my baby HLA, human MHC. These three are the ones that I would want to investigate. Oh, chromatin. So chromatin, so your DNA is wound up in your cells. You have a lot of DNA, and to pack that DNA, it's packed around things called histones. So histones are made of this protein called chromatin. And so chromatin remodeling is the process by which your DNA like winds and unwinds, allowing portions of the regions to be accessed by things like RNA polymerase to be turned on or off. You can imagine like your DNA is very wound up very tightly, but like locally it has to kind of unwind for stuff to happen. And so that's what chromatin remodeling is. I'm not like a DNA expert though, so correct me if I'm wrong on that. So combined with the other previously mentioned genes, it's really hard to put it all together. Like I hope you are getting an appreciation for how stupid complicated schizophrenia can be, right? Not only are there like random things that are associated like tRNA processing, chromatin remodeling, they might not be random, right? But it's like hard to figure out what the connection between those things and schizophrenia would be. I hated biology in high school because teacher gave you the impression it was all like zoology. Yeah, but zoology I think is more popular for more people, so I definitely get why that would be in that way. The crazy origami that DNA goes through blows my mind. Wait till we see the triple helix stream. DNA is an incredibly complicated and powerful molecule, including having the ability to be triple stranded, which is just nuts. Yeah, triple stranded, which is the main plot line for a wonderful show called Zoo, where triple helical DNA causes animals to start becoming hyper intelligent and killing humans. It's of course that's not what triple helical DNA does, but when I was watching that show, I was like, there's no way that triple helical DNA does exist, right? And then I looked it up, and lo and behold, DNA can be triple helical. That's nuts. Zip and unzip version of DNA. You need to unzip a file to use it. Exactly, dude. Chat is smart today. I mean, not to say that chat isn't normally smart, but chat is really smart today. That's oh baby, it's a triple. Yeah, true, exactly. Oh baby, it's a triple. Yeah. So all of these different genes make it really hard to come up with one reason schizophrenia happens. So where are we now, and why am I kind of hopeful for the future? So correct me if I'm wrong, because I'm not fully well read on the latest and greatest in schizophrenia treatment, but we have this class of dopamine antagonists that are antipsychotics. And as I learned from Toxo, there are a second generation that also have serotonin stuff in there too. So that's good that people with schizophrenia are getting more effective treatments that help more different parts of that condition. Schizophrenia ranges in intensity, so does responsiveness to treatment. So it's not always doom and gloom to get a diagnosis of schizophrenia, but it is doom enough for society to invest in trying to fix this as an issue. Regulating dopamine treats the psychosis, but I want us to imagine a future where we can prevent and treat it before the psychosis occurs. I think the first instance by which people have their first psychotic episode when they develop schizophrenia is actually quite early. So you can imagine, 22 having a psychotic break and then knowing that there is a chance, it's not always the case, but there is a chance that you have this reoccurring in the future. Like, to me, that I would like for that to not be the reality for people anymore, right? That's the best way that I can phrase that. And part of making that a reality is understanding the basic biology of schizophrenia as much as we can. So even though it is polygenic, even though there are 100 plus, there's at least 100 different associated SNPs with schizophrenia. And even though that system is so complicated, I want to go back to an earlier figure. Even though this part, right? This part up here, right? The genetics, even though this part is really complicated, we can break it down one portion at a time. And if we can really nail down the pathophysiology, we might not have to know everything about every gene, but if we know enough about any of these other things, it might open a path for us to make sure that people don't have to live with this thing. I don't know if that's rude to say, but this condition that can separate them from experiencing reality, which I think is worthy to do, a worthwhile thing to study, and something that I didn't do in graduate school. My actual training was in brain injury, traumatic brain injury, because, I am personally very moved by conditions that rob you of your experience of being you, right? So traumatic brain injury is another thing that kind of can take elements of who you are away. So for me, these are my important issues, the ones that I think about the most. Yeah. So I'll go back to reading chat for a bit while I grandstood, soapboxed about my particular research interests. How does that even, isn't double helix because ATC forms binary associations with each other? Yeah, but if you bend the backbone in a little bit of a different way, you can actually get triples, and you can actually find images of those online. You can actually just Google triple helical DNA and see that happen. I don't really, it's like there's like Watson-Crick base pairing, and then there's like Hodgstein some other dude base pairing where the configuration and the orientation of the base pairs are slightly different, and the Hodgstein version I think allows you to cram in another axis. There's also double-stranded RNA, which isn't quite as cursed sounding as triple, but it's sort of weird to think about at first. Yeah, you typically don't think about RNA as double-stranded unless it's out there destroying stuff. It's prevention a cure. You could call it a pre-cure. Yeah, true. Pre-cure. Very cultured reference. Yeah. Yeah, I hope you enjoyed what I had to say about genetics. I hope you got an appreciation for the very basics of how traits are inherited. Now, the other thing that I wanted to distinguish just as like an interesting idea is that when people say heritable, technically that doesn't necessarily have to be genetic, right? Behavior is heritable because you have parents who model behavior. So I just wanted to throw that out, that not everything that is heritable is genetic. You can learn and inherit things from your parents that are beyond your DNA, which is why, like, as a biologist, I want to be very careful to separate myself from the idea of biological essentialism as well as I can. Are triple-stranded found in any living systems? I think, if I remember correctly, there is one instance of triple helical DNA having a inhibitory function. I don't know if that was in humans, though. I'd have to check. It's something that you can get to happen in a dish for sure. I think I read about one instance of it being actually functional, but I'm not entirely confident on that. So I'll take the time right now to field any questions that you have. I realize I don't do that often enough in my streams, just to pause at the end, let you stew in what we talked about, and I might not be able to answer them, especially because I am very ambitious in doing this 101 series. You know, I'm not a schizophrenia expert nor a genetics expert, but I think it's fun to talk about biology. It's so fun. Chat is popping off today. It's been really neat. Also, I think while we're talking, it's ending, I can chat for a bit longer. While people are thinking about questions, I'm going to tab up and kind of collect some of the names that I saw earlier for this cat. So, cat. And we have polymerase, right? Polymerase. Polymeourase. Those are all so good. I don't know if there were any more, but those are really good. You were collecting names for a cat? Yeah, my very own cat, who you sometimes hear get fed when it's 5 o'clock EST and can meow when she wants attention. Purkinje, Nyakrof. Dude, these are all so good. Purkinje. Oh, wow. Wow. Those are really fun. That's great. I think macrophage is a little hard to say. Mitochondria. Ferber house of the cell. Oh. Oh, man. That's so funny. Those are so good. Oh man, I don't know if I can Purkinje really, I think Purkinje is very close to my heart because of my background in neurobiology. Felinocyte. Felino or Felina for short. That's really cute too. Permalation, not meowization. Dude, gotta write these down for merch ideas. How big of a YouTuber do you think I have to be to roll out merch? I almost called it meowch. Getting pilled by chat. Yeah. So, yeah, so I guess I can talk a little bit about the collab next week with Ressa, if for those of you who maybe join the stream later. But I have a stream next week on bioinformatics, next week, 2 p.m. Saturday, with Ressa Makurabe, and we're going to be using programming to kill bacteria, or to find strategies to kill bacteria, which is going to be very fun. So if you're on the programming end of things, I think learning how your tool is helping biologists think through how to tackle things like antibiotic resistance is going to be really, really fun. And also Resta's just a great streamer as well. Very relaxing kind of voice. Meowosis, that's great too. Meowosis. Meowtosis. I want more cats to give them more of these names. 2 p.m. EST, correct. Do it, make merch. Eventually, I think that would be a very fun goal for me to have. I'll have to dredge the severely atrophied bioinformatics parts of my brain. It's not as atrophied as mine, considering I dropped out of bioinformatics. Staphylocactus. That's good. More Cassie say Tox is very happy with that. Listen, when we actually move to a place that has more room than our, tiny apartment, I will gladly have more cats. At least 500k subs for merch. I trust you will definitely reach it. I talk about this often on stream, just because I want to be honest about who I am as a person. Like, I'm very confident. I find that so fun because, not funny, but like, there are a lot of people who struggle with imposter syndrome, and I have the opposite. I have poster syndrome. I don't know. I feel like I will make it. Thinking and feeling are different, which is why I'm careful to separate those two ideas, because thinking of like 500k would put me into the range of a big YouTuber, or like a big enough YouTuber, not 1 mil, but certainly bigger than I could ever have imagined this channel becoming. But I feel I could do it. I have the inherent feeling that I could do it. It's not motivated by anything logical. I just feel like I could do it. Of course, not without the help of my very generous and helpful community, right? But I think, I don't know. I feel like I don't want to lie about who I am to garner sympathy. Not that I think that anybody would do that, right? But like, I don't feel the need to hide that, bros, we're gonna make it. We're gonna make 500k. I don't know when, but we're gonna do it. We're gonna do it together. It's gonna be really fun. You have the opposite of what? Imposter syndrome. Yeah, poster syndrome. I'm very poster. Powerful. I'm a powerful poster. I think I can do everything, and I'm often hard checked, as Dr. Wendell on that neurobiology stream can attest to, because I always feel like I can do anything, and then that stream checked me hard. I actually have to do programming for my job now, to program something for the students, and let me tell you, I was very confident going in, and now I'm in misery, because again, I am hard checked by reality constantly, but that never stops me from feeling like I believe in myself. And I really wish I could tell everybody with imposter syndrome like how to do that, but I honestly can't. I'm just, I don't know, dumbly naive and optimistic, or maybe I failed so often in my life that failure doesn't really bother me anymore. It's an interesting topic, I think. Welcome to the stream, Saikime. Welcome. Unfortunately, we're nearing the end of the stream. You missed a pretty meaty stream of smart chatters brainstorming the biology behind schizophrenia. Very fun. Fake it till you make it. The thing is I'm not even aware that I'm faking it. Like, I just think that I'm awesome, which is really cocky. But and to be honest, to be clear, feeling and thinking is different, right? I feel like I'm great, and I think I'm good. and those two internal forces are constantly in constant battle with one another, of my super ego, putting my ego in check, if you would. The Freudian super ego, putting my normally what we would call ego in check. I find your videos more entertaining than some other larger channels. You just need the algorithm to pick you up. Yeah, I think part of I think the downside of my channel is I'm not very good at thumbnails or titles. I'm getting better for sure, and Tox is of course very helpful in designing these thumbnails. But I don't know. I'm my visual IQ is very weak, and I think that is difficult for the algorithm to pick up. Welcome to the T cells, Brandon. Thank you for joining the membership. Thank you so much. Yeah, that's awesome. As a member, you get perks like deciding what I cover in a future journal club. Because this is a part-time thing, I don't really have a guaranteed schedule on all of that or anything. But the next member suggested stream I'm doing is about the brain virome/bacteriome, which I think is a member. It's definitely a member suggested topic, but I think it was may have been iCausahesion who suggested it, but we're finally going to get to that at some point in September. Yeah. Yeah. What language? Python. I love Python, but the fact that I think Python is easy makes it so that when I actually have to program, I realize that Python, the language, is easy. Programming is not easy. And again, checking the ego. I said it before, hell respect. Thank you. Thank you so much. Share some of that confidence with you all. Maybe I should just do a stream where I say nice things about you all. You know, I haven't said that I'm proud of chat today. I've called you all smart, but I would like to emphasize repeatedly that I am very proud of how far I'm able to push chat, and I'm loving the because to me, what was really important to me in my development as a scientist is really internalizing correlation and causation, like really getting it, like not just shit posting on Reddit thinking I'm smarter than everybody on our science, but actually understanding the nuance of correlation and causation. Not that correlation is always useless, but that correlation has a role to play in the larger story. And the fact that I think from the polling that I'm doing, the fact that you are picking up on that means that I believe that I am achieving my objective with these streams. Because even though these streams will never help me with the algorithm, but almost more important than the algorithm for me is that people who want the chance to study like a scientist are given that opportunity without having to pay to go to university. And I think that's really special, I think. Don't just put Fi's face with his mouth wide open in a thumbnail. I actually did. It was one of my lamentations to talk so early on in my YouTube career. I'm like, the weakness of Fi is that Fi can't pog. Because if Fi could pog, think about those thumbnails. But I don't think I need to pog in the thumbnail anymore. I've grown past that idea. I didn't realize it would blast me up like that. No, members should be acknowledged because you are supporting the Toxo Omakase Fund, but also the idea that there can be content like this on YouTube for people who are looking for more than the basics. I'm not saying I'm the only one, but I think the algorithm is less friendly to the deep dive people. So any support that you can show is highly, highly appreciated. You can change the titles and thumbnails after posting the video. Yeah, A/B testing, which I'm going to be doing for the pre-on video, just in a little bit, playing around with. Because again, with the whole I'm super cocky, I told Toxo repeatedly, I'm gonna make the best pre-on video on YouTube. I'm confident. I feel like that my video is the best pre-on video on YouTube. And again, I feel that way. Do I think that way? No, because the numbers don't reflect it. The algorithm didn't really care. I mean, it cared a little bit. I wouldn't call it a failure, right? But my expectations for that video is that it does as well, if not better than the rabies one, and it didn't. Is that disappointing? Not really, because my brain is keeping my heart in check. My brain is like, hey, let's not be cocky here. You don't deserve or need 300,000 views on that video. But my heart is like, I'm awesome. This pre-on video is great. I'm really happy with how it came out. And I am. I still feel like it's the best pre-on video. At the very least, it's the best pre-on video I could have made. And I'm very proud of that scripting. I'm glad I found your channel. Thank you, thank you. At the part about risk factors, I think psoriasis is the autoimmune disease most associated with schizophrenia. Really, HLA. So that's another thing that's associated with HLA. That's interesting. Drawing a pogging phi does sound fun, though. I feel like it'd be so cursed. Actually, I am curious. I do these every once in a while because I do want to know where my streaming audience comes from, because it's pretty easy to know where my video audience comes from because you all come from the algorithm. But the people who choose to stay, I'm always curious as how do you find this channel? And then, because I do a lot of my own PR, and I feel like it doesn't really catch. I think the algorithm is my best friend here. The Prion stuff is what made me sub. Yeah, sorry for those of you who want to do Nalgeria fowleri, right? Brain-eating amoeba. I'm just going to be very transparent with my streaming audience because I feel like you're somewhat, at the very least, invested in the health of this channel, and I want to do one big video a year. This year it was rabies and prion, but I feel like I needed the extra momentum right off the gate. Everybody's been clamoring for brain-eating amoeba. Everybody's been clamoring for the Black Plague, but I don't want to do them too soon because I want to do them slowly over time and show you all the other cool things in biology, so you don't just show up for the cool pathogens that you like and then immediately leave. I want to like pill people I want people to be bio-pilled. It took me long enough to actually understand correlation causation. At first I thought I did, but I didn't. Exactly, that was my experience too. Like, you have to really practice mindfulness with regards to scientific practice for you to actually like internalize that idea, because it's easy to understand it when you first read it. To implement it in your daily practice of thought is different, and it can crop up in all sorts of different unexpected ways. Did I ever tell you that Sai introduced me to you? I think I heard about that. Yeah, me and Sai, we have a bunch of overlap in terms of, well, obviously we're both part of V-Theorem, but like our mission is very similar: to bring high-level academic content, make it accessible and engaging for an audience. Because the thing is that with tardigrade, this was like my most frustrating bit with the tardigrade stuff, is when you watch tardigrade videos on YouTube, it's very entry level, which is great, and that's why they do well, because anybody can watch one of those videos. But my attempt is, I want to start in a place where everybody can start and end in a place that I find interesting, right? Tardigrades can survive all sorts of terrible things being done to them. That's cool. Don't get me wrong, that's cool. What I find interesting is why and what molecules are involved. And so that's kind of the goal here. Would you consider proteins like alpha-synuclein or amyloid beta as prions? I'm not in that. I purposely avoided this question in the prion stream because I know how contentious that idea is. But just as a reminder, as More Juice has correctly emphasized my mantra, biology is a spectrum. So functionally, what does it matter? Why is it important that we classify alpha-synuclein or amyloid beta as prion? Does it actually change anything? I don't think so. You're still going to study amyloid beta and alpha-synuclein as their own independent proteins. Drawing the comparison to prion protein, just say they're prion-like. Like, it doesn't actually change how we think about them. And so those labels are not very interesting to me or important. I like to listen to scientific videos when I'm driving to and from work, as well as when I'm home gaming. Yeah. And I think that the stuff that really makes you think, it really makes you think, is the stuff that like I went to grad school for, and I like, you shouldn't have to go to grad school to flex your brain in that kind of fun way. Yeah, 94% from the YouTube algorithm. Yeah, I kind of figured that. Unlike a lot of other VTubers, I think my success is always going to come from the algorithm and not from my own personal branding, which, you know, sometimes I feel like I should give up entirely, but you never know when your stuff gets picked up by Twitter or Reddit or anything. Yeah, YouTube algorithm. Yeah, I understand. Don't exhaust yourself from doing too many big projects. Yeah, I've been better about giving myself more time for these things. Although I want to maintain the reputation as someone who meets deadlines, because that's just the person I am. I set deadlines and I meet them. But giving myself some space to do that is important. I've been getting better at that. Found you via Plaguing Shorts. Toxo has been saying that I should get back on the shorts game. I don't think you all know how depressing it is to see how dumb some of them. Sorry. I shouldn't. But if they're putting hate comments in my comments, they're not watching my other comment anyway. But I've been very reluctant to get back into making shorts because my stupidest comments come from the Plaguing Shorts. And I don't like reading them. But Toxo's right. It would be good to do more of the shorts. They are more algorithm friendly, and she has a really good idea for a series of shorts that actually will help me apply for a grant so that I could join Psy as being in the very exclusive group of VTubers to win official approval from a body of researchers. Thank you so much. This is a very nice streamer love kind of talk, and I do appreciate the kind words. Yeah, referring to icosahedron. Yeah. And to see the art that came out of to the animation that you made for that I posted in the pre-on video, I could not stop talking about that. That actually made me so happy. It was amazing to see. And it's part like, it's things like those that, like, I don't rely too much on external validation. But it's things like that that help the momentum, right? That makes it like, oh, what I'm doing is good enough that someone would go out there and animate an amazing thing, which is like external proof. And that contributes to me being like, the feeling that, like, we're gonna hit 500K, we're gonna hit 1 million at some point, because I don't see myself giving this up anytime soon. And your support has all been very important, of course. Work-life balance as an academic, is this real life? I love my job. My job allows me to make content. I would fight anybody. I would get into a knife fight with anybody to keep my job, because not only do I have income, and not only do I get to teach for a living, which I love doing, I get to then do this as a hobby, and then earn YouTube ad revenue to buy myself a computer that actually lets me stream. It's been pretty incredible. Thanks for dropping in, Luke. Good. Have a good dinner now. Work-life balance as an academic, is this real life? Yeah. I did it. Although Toxo can argue that there are points in the school semester where that does not appear to be true for me. But I'd say for the most part, I do love my job, and I love being able to do this. I have shorts disabled on YouTube. I get that. I felt myself getting TikTokified. But eventually I just started accepting that shorts are always going to be a part of YouTube. Yeah, congrats to Sai. Yeah, it's really incredible. It's really inspiring. It's not that I'm jealous or anything, right? Baka. It's not that I'm jealous, but I am chomping at the bit. There are 3 deadlines for grants that are coming up in the year, at the end of this year, and at the beginning of next year, that I can't wait to apply for. Like, my dream for the streaming aspect of my YouTube channel is, it would be really cool to have a V-Theorem live seminar panel thing about education in VTubing, and to be able to do that for an audience, like, to imagine the future in which I am that popular, supported by funding from researchers who believe in what I do. Like that, I'm prone to dreaming big, but I also never stop working. So one day that would happen. Yeah, the animation was really dope. As someone who's trying to dip their toes in pixel art for game dev, it was, oh, do you do game dev as a hobby? That's really fun. Although I hear it can be quite nasty. I am living the dream. What computer did I get? I don't even know how to say all the numbers. Although in the Discord, there's the full build as my members were trying to help me troubleshoot. You got this, get those grants. I will do my best, says the brain, and my heart says, I'm gonna get them. I'm gonna get them. Nothing's gonna stop me from getting them. And my brain has a, can't, you know, hold me back and be like, there's a chance you won't get them. You know that, right? But that's the thing. That's a very bad attitude to have as a researcher, because you are always disappointed and things never work when you're a researcher. So it's constant failure. But now that I'm not a researcher, things work out more than 1% of the time now that I'm not in graduate school anymore. And that's been really, maybe that's why I'm so confident that when you fail daily, hour to hour as a graduate student researcher, it makes you more appreciative for the success that you do see. And maybe I'm unlearning the trauma from graduate school as a Psyche-May you can attest to. Psyche-May, by the way, another VTuber like Kuro. Psyche-May is a psychologist VTuber. You can follow her on YouTube. She's done some fun things about analyzing the ethics. I think the ethics in Dungeon Meshi, right? I haven't tuned into that because I haven't watched Dungeon Meshi, but that sounds like a very fun and exciting topic to have covered. I wanted to clip that, but forgot this is YouTube. YouTube does have a clip function, but I actually don't know how to get to it. Yeah, V-Theorem panel and something like Anime Expo would be crazy. I think Anime Expo, or what is it? The other one that's specifically for VTubers. I've forgotten. I've forgotten what that one was. Yeah. VidCon? I don't know that it was VidCon either. Resta went to it recently. I don't remember. Offkai. Thank you, Psyche-May. Yeah, Offkai. I think Offkai would be a worthy place. Worthy, like it has to be worthy of me. I mean, like, it would be appropriate for a VTuber to have a panel, like a smaller VTuber to have a panel at Offkai than it would be for a VTuber to have a small VTuber to have a panel at Anime Expo. Wouldn't say I do game dev as a hobby, at least not yet. I'm still learning. That's the other thing that like programming really helps you to enable you to do, is that like I've been running D&D campaigns on and off since I was a high schooler because I like the idea of game design, but none of the patience. Although for my programming final as an undergrad, for my Python programming final, I programmed a little game that you could play where you would have to treat patients. And I used like a South Park image generator to generate all the characters so that I wouldn't have to buy art as a poor college student. And I got a B+ for that project. It was okay. At the time, I really wanted to be a doctor, so I figured that doing something medicine related would have been appropriate. But yeah, as a dungeon master, you really do get to have all your hands on all the different levers of game design without ever having to run into a programming error, which I think is the best of both worlds. But I talked about this in my Discord. For those of you who want access to the Discord, let me get a link so that I'm going to do it in batches, and then when I'm comfortable enough, I will make it public by posting on my YouTube. But for now, let's try to get the invite link so I can expand this Discord community. Why can't I make an invite? Bear with me for a second. No, why can't I find the thing that lets you invite? It's saying that I have no active invites. Oh, how does Discord work, man? What a trouble. That sneaky link, yeah. This is so sad. I don't know how to invite. I thought I had the thing up, but it won't let me invite. Am I just dumb? Am I just silly? I might just be silly. Maybe I'll try again in a little bit, or maybe I'll just make it public after today's stream. I've been pussyfooting around it long enough. Just finished exams, so I'm open to trying new stuff. Well, until I have free time. You said you were at a med school. What year are you in? Thank you, Toxo. I thought I understood, but I don't know. Do we get a pre-order bonus for joining before this channel gets big? Well, what does pre-order bonus mean? I will say that the fewer T-cell members there are, the more chance your idea of what I want to cover gets covered, even though I've not been very strict on when I decide to cover those. Soon to be second year, hopefully. So you just finished your school-heavy year of medical school. Dang, you do medical school and you do all that art? It's like, it's bonkers. That's actually nuts. I had forgotten that you were in medical school. That's good. Your first year being over is probably a really big relief. And then you get to do more doctor-esque stuff as you go on, which I believe will be fun for some people, but for me it would have been really bad. I don't think I would have enjoyed that. That's a shutdown memberships need to be exclusive. True. I mean, I'm still waiting for people to subscribe at those insane higher tiers, although I might actually end up removing them because having rolled out membership, I can barely do the T-cell membership ones. So I think the learner challenges are a little ambitious. And since no one has subscribed up there, it's actually easy to delete those membership tiers. Being a GM for a D&D campaign automatically grants you uber nerd status. I think it grants me uber geek status because I think nerds and geeks are slightly different. It's a very geeky hobby. I wouldn't call it a nerdy hobby. Also, welcome to the stream, Ferrum. Yeah, we have a whole art club in my university. How common is it for medical students to have extracurricular lives like that? That's surprising to me. Because medical school, part of why I was like, I would not function well as a doctor, not only because the job itself is very stressful and you have to deal with so many people, but also the volume of learning that you have to do in that short amount of time is impressive. And so you have all the, again, I dunked on Psychen a lot because I was like, all doctors are freaks repeatedly in that stream. But I do have a lot of respect for people who can make it through. It's not easy. Yeah. Right. Yeah. Here's something interesting that I think would be worth talking about. I don't know, or maybe should I save it for other streams, considering it's getting kind of late. Oh, but I'm having fun. But it is getting kind of late. I guess I can plug one more perk that I have for members is that I was thinking I was going to do more gaming over the summer, but then my job happened and the pre-on video took a lot longer than I expected. But like, I will be doing a gaming thing in August with my members just because it's important to work, but it's also important to rest and it's also important to play. So there's that in terms of a membership perk. I'm sorry, I don't offer much for membership perks, except for being able to guide the discussion. Although if in whatever universe this becomes my full-time job, that's bound to change, obviously. But, you know, the support is appreciated. Hey, don't scare off future doctors. Actually, I think it's not me that's going to be scaring that off. It's the system. It's the system. It won't be me. I don't really know what you are referring to, Kuro, in terms of teaching. Oh, in terms of medical school? Yeah. That's probably a question for icosahedron, because I do not have any tips for medical school because I decided to not. Will I get the sub then? You are my favorite YouTuber. I did say that I would. I don't want to show favoritism to certain chatters. I think that crosses a ethical boundary for me, but I will honor the fact that you did get the immunity connection with schizophrenia. So I will buy one membership for chat and I will let the YouTube gods decide who gets that. So drum roll, who gets gifted? Oh, my security code. Let me read all of my credit card information out loud. Oh, but then I have to enter a whole different card though. Not today, not today. Not today. Okay. Let's gift a membership. Who is it going to be for? Sorry, kind of... who gets that? Where is it? The Wolf was gifted a membership. Welcome, welcome. I still can't believe YouTube just doesn't let streamers give membership to specific people. Some streamers can do that. I don't know, but like Faye Dressera, who's also a VTuber, who also does educational content, she can give away subs to free. I don't know why I can't. I keep on forgetting to call YouTube support about that. So, I don't know. YouTube seems to be playing favorites. My hobbies in med school were having chronic arthritis. I'm so sorry about that. Arthritis, I believe another list of things on the autoimmune list. May I be a psionic tabaxi in your game? I tend to play with the more vanilla D&D stuff because it just gets so wet and wild the more expansion stuff you get to that it gets kind of hard to get everything in my head. But that sounds very fun for sure. Yeah. I mean, no, I can't. I can't start another D&D campaign. I was like, it would be fun to play D&D with chat. It would be fun. No, I can't. I already have a D&D game going. This is the adult me. This is the super ego being like, you cannot have another creative thing on your plate. It would be unwise because if you have too many things on your plate, then everything else suffers. To be fair, to be honest, I've been looking into Pathfinder. I have two. Pathfinder, because the combat in Pathfinder is more interesting, presumably because the combat in D&D is really boring. I bet chat is way cooler than your other campaign. I don't know, my other campaign, I'm very blessed to have a full table of people that are very enthusiastic to play. Because in my experience, sometimes you put together a D&D group and they just kind of view it as like a way to hang out and people are on their phones and, you know, it's just kind of whatever. But the group that I have now, they're like actively invested in the story. They have their own interactions amongst... Oh, I want to tell a story. I want to tell a story about our campaign. The extent by which I'm so happy with the players that I have is that like... So the players that I have, they're in the campaign, and right now there are like interpersonal drama between the characters. And as a DM, I don't make drama for the players. I don't like pit them against one another because my philosophy is that if people are cooperating, it's more fun. It's more fun to play D&D as a cooperative game and not a game where you backstab each other. But my players are so dedicated to their lore and so dedicated to their character that they will very faithfully roleplay interactions that are negative. Like if they disagree, they will disagree and they will not just be like, oh, we're playing a game, let's just agree to move on and you know, whatever. They will hash it out at the table because they're that committed to role playing their specific characters. And it's like, on one hand, it's terrifying as a DM because you're like, oh no, the people are fighting. That's like, that means they're not having fun. It's stressful. But like, they've assured me that they are having fun, that these interpersonal conflicts, this process by which they come together and solve these problems that are still faithful to their characters is part of the fun for them. I'm like, damn, I really lucked out with my campaign. Role playing, I think, is the fun part of 5e because combat honestly kind of sucks. I have loved D&D so much, but I have no friends to invite, so I just kind of join when I can. It's going to be that way for a while. I got really lucky. You just got to keep on trying, just like this YouTube channel, right? You just got to keep on trying. Eventually something will happen. What games do I play? This is my first stream, so I'm sure you've mentioned it before. So my top games are going to be RTS games, strategy games in general. I have over 1,000 hours in Civ 6. I've loved Warcraft. Sorry, Warcraft 3, not World of Warcraft. Warcraft 3, StarCraft. Those are the those are the games that are most important to me as a gamer. That and Slay the Spire. Undertale was enormously impactful on my development as an artist, even though I mean, I consider my YouTube channel art, but the way I view art has been severely impacted by Undertale, as cheesy and cringe as that might be. But you know, you can't argue Undertale is good. I played a lot of Pandemic as a kid, which is why I thought to do the Plague Inc. series, which, by the way, legally distinct. You're gonna get mad if you confuse them with one another. Baldur's Gate Descent into Avernus. Actually, no. I don't actually play RPGs after Undertale. Undertale killed the entire genre for me. I mentioned this in my Discord. Like, Undertale and Slay the Spire combined killed my enjoyment of RPGs, which is sad in a way, but I still play video games. It's not like it ruined video games for me, but just RPGs as a genre. I think you're doing something right if as a DM, if people feel comfortable, immersed enough to fight. Yeah, and to ensure that they're still having fun, right? Like, it's not just me having some measure of success. It's the fact that I lucked out in getting a group of people who are dedicated to the idea of roleplay. That is truly a blessing, because it's you don't, because the thing is I have such jealousy of professional artists, and I know it's a grass is greener type situation because I have a stable income. And I would never trade my life for that of a starving artist, except maybe if YouTube really pops off for some reason. And I know it's a grass is greener type thing, but the ability to make art and then to have people enjoy that art is extremely enjoyable. Profoundly enjoyable. And to write like my own edgy fantasy, high fantasy, cryptic, spooky kind of story, and then have people have fun in that world, like, I would never be able to write that book because I'm not a good writer. But D&D is an art form where you don't have to be a great writer, a great artist to have fun. You need two ingredients. You need to want to do it and put in the work, and you need people to buy in. And as long as those two things are true, you don't have to write a Game of Thrones level epic battle thing. You can write what you want to write, and what I want to write is fan fiction for the world that I made up. And of course, if I were to publish my D&D campaign as a module, nobody would buy it because I'm not good enough. But you don't have to be that good of a DM to have fun playing D&D. And that's the overall harm, I think, that DMs like Brendan Lee Mulligan and Matt Mercer have done damage to the D&D world, because they're too good. They set the bar too high for the casual enjoyer. So if you want to play D&D, just do it. Find some people, give it a try. They might not like it. You might have the campaign end, but just do it. Just try it. It's so much fun. Chat plays series or something. I don't think I'm going to play D&D with chat. I think it's way too much prep. Maybe next summer. I could set an incentive for next summer to play D&D with chat in this like gaming era of Fi right before classes start. I think that would be fun. I played a ton of Plague Inc. when I was much younger, sort of inspired my initial love of medicine. Yeah, me too. Gaming is actually, gaming is art. It is art. It's interactive art that you can experience. So that's very pog. The change in conversation from genetics to D&D, amazing. I mean, there are a lot of another really great D&D group I met in graduate school. Nerds are gonna nerd, you know, commonalities and that sort of thing. Being a pro artist sucks because you have to make what other people want instead of what you want to make a sufficient income. Art as a hobby is the best. That's also true. To be dependent on your art is maybe not as fun. The best case is if the stars align and you get to make the art that you want to make. I think that's the life. That's honestly aspirational for people who need to have passion behind their work, right? Because I talked about this with my office mate. I would do terribly at a job that I didn't love, right? If I had a job that I didn't hate, I would actually not be very good at it. I would only be good at jobs that I love doing, which is why I'm not going to be a benchtop scientist. I didn't love doing science, but I loved teaching. And so for people who have to love what they do, that's got to be the dream right there. Factorio, I don't think I've played. Ever played Rebel Inc.? No, I don't think I've played Rebel Inc. Subnautica biology. The thing is I thought these are all, so Subnautica was on the list of things for me to do, but since I've debuted, there have been other VTubers that I've discovered. They may have debuted after me or before me, but there's more and more VTubers that are entering the education space, and so I feel like I'm no longer the biology VTuber. There are plenty more of other people who do biology. I was never the biology VTuber. But at the time, for the penetrance that this kind of stuff has in the market, I didn't know of any other biology VTubers, and now I do. So I would not feel comfortable making commentary on animals as species. For that stuff, there are other VTubers in that space, one of which I may or may not be collaborating with. I don't really know. It's kind of tough to work out scheduling, because my language is molecules and cells, and animals are complicated. One Shot got similar vibes to Undertale. I've not heard of that, but if it's got Undertale vibes, I'm likely to try at the very least. D&D chat campaign with multiple choice. Exactly. That's, I think, a very fun idea. Yeah, like maybe 4 votable options, and then if there's a juicy chat suggestion, then like we can do that one, and I think that would be quite fun. The issue would be tempo, because D&D is a very slow game. I mean, when I think about it, if I do a summer gaming stream, because I'm planning to do like a gaming marathon before classes start, a marathon D&D session with chat for like a good chunk of like 8 hours or something, like that sounds pretty fun, actually. Neutrophil Identity Crisis arc. I don't really know what you are referring to there. Glad to see more people getting into chemistry. Biology on YouTube. Chemistry has had a monopoly on academics on, yeah, like Nile Red and Minute Physics for Physics. Like this was exactly why I started YouTubing in general, because I identified what I perceive to be an unexplored niche, and that is popular biology content for entertainment. Because SciShow is for education, Khan Academy is for education, and Nile Red is for entertainment. Nile Red showing you how to make gloves into grape juice. That's awesome. That's really fun, and it helps people stay interested in chemistry, but it's not for education. And I wanted to enter into that space because I was like, well, no one's doing that yet, and not in the exact same way that I would want to do it. And I'm happy where I'm at, because being the VTuber that I am, I actually had a much more vanilla version of this video or this channel before Toxo made all this art and rigged Fi. I was going to be a dude in front of a camera, and I'm so happy that I'm not, actually. If you count food science as biology, then biology also has long ago taken over YouTube. I guess, although there's been kind of discourse about the longevity of cooking channels on YouTube or food science channels on YouTube. I think that's interesting, is whether or not my content has longevity. My argument is that there's only so many recipes. I mean, that's not true. In terms of like my goal for retaining you, my audience, my hope, my plan, my conceit, my ethos is that if I pill you into loving biology, then I get to keep you as an audience member forever. Instead of cooking channels where, like, I used to watch Chef John every day, but eventually, you just cook everything. And to me, that's not very appealing anymore. It's lost some of the novelty. And I love Chef John, don't get me wrong. But in terms of, am I a regular viewer of him still? Not really. I'll go back every once in a while if there's an appealing idea of a dish that he has. But if I pill you into loving biology, then the goal for me is simple. There's so much interesting crap out there that I will die before I get to cover it all. And so I'll get to hang on to you forever. Phi Yandere. The Amoeba Sisters is also, I would rank that in education and not entertainment. They are also similarly cartoony, but they are very much for the goal of helping people who are in school, at least from my perspective. Because, I mean, my particular angle is that You should not watch me to get better at school. You should watch me to stay interested in biology. That's the goal. You're not going to do better on your... Although there were some other chatters, and believe me, I really do appreciate those comments, but I don't structure my content in a way where my goal is to help you get a better grade. Because if I were to do, and that's why I like this format of my lectures, quote unquote, like struggle to call them a lecture. The structure of the stream is so that, like, if I wanted you to do better in genetics, I would have made you do the 4x4 Punnett square. I would have made you do that live because that's what gets you a better grade. No, what I want to do is keep you engaged, to keep you hooked, to keep you wanting to learn forever because learning is awesome. Thought Emporium? Oh, we don't say the name of that channel on this. We don't say the name of that channel on this channel, Aiden. I think you're maybe a newer chatter, but yeah, we don't. It's like Voldemort. Yeah, I did get triggered. Yeah, the Thought Emporium. Let's just say, Aiden, if you go back into my streaming archive, I've done a whole stream where I hypothetically analyzed a hypothetical video which someone hypothetically tried to hypothetically cure their own lactose intolerance hypothetically and did so in a very irresponsible way. And that channel, it may or may not be the Voldemort channel. I've never been so focused on 1 hour long video of mixing chemicals until I stumbled upon Nile Red. Yeah, Nile Red is... I love Nile Red. I've watched every one of those videos, and they're incredible. And I think it'd be cool to meet him one day, just because that dude is fun. It also makes chemistry accessible, right? Because I think I'm more similar to Nile Red in the sense where, like, is it that you're going to do better in class? No, but you're going to see that chemistry is accessible, that what he's doing, you know, I could invert a flask. It doesn't take a genius to invert a separation funnel, right? And so it makes it seem like you can do it, and you can. And I think that's valuable. I'm out of the loop. Why do we not talk about him? Chatters. I don't want a clip of me circulating on the internet of me saying all those words in the exact order that I want to say them in. So I'm sure one of the chatters might be able to help with that. It was one specific video. The rest of their content is still good. I have not watched the rest of their content, although I believe I'll accept that from More Juice, that, you know, just because you have one bad video doesn't mean that the rest of them are bad. Although I do believe that one is pretty bad. Separation funnel is triggering my fight or flight. No, believe me, like, Nile Red has repaired my relationship with experimental chemistry, because I also had quite a lot of difficulty with chemistry in class. Yeah, I hypothetically tore into one of those experiments, and I hypothetically came to the conclusion that they hypothetically never had lactose intolerance to begin with, but rather some other GI condition that looks like lactose intolerance that they were misdiagnosing, because the things that all had to be true for their experiment to work did not seem very likely, hypothetically, of course. I don't want to start beef with, you know, this channel in particular, which is why I thought long and hard about doing that as a full 5 video, because I was like, ooh, you know what people love on YouTube? Drama. But I don't want to be a drama YouTuber. But at the same time, no one called that hypothetical channel out. It's just allowed to exist with no pushback, and I think that's ridiculous. If any journalist wants to copy any of my points exactly and not give me credit for it, go ahead. I don't care. I don't want to get famous off the back of drama, but I did want my audience to appreciate what a potential load of potential bull honky that whole process was. I want to avoid the iosaur. That's a very good way of putting it. That's very true. There definitely exists lactose-tolerant milk intolerance. There's like, it's not entirely clear what this hypothetical person had. But again, watch the stream. I'm really proud of the conversations that we've had in that stream, because it's an interesting thought experiment to walk through science that can hide a lot of detail, and why you need professional hyperfixators to challenge everything. Like, science is not for the weak of heart, in the sense where, like, what I love of everybody on science, of course, but it's not appropriate for everybody, because you have to be obsessive with your relationship to the truth. Because if you are anything less than obsessive with the truth, then you end up doing subpar science. So that's my spiel on that. I think accountability is important. What drives me crazy about this hypothetical video is that there is a very simple, very simple molecular test for lactose intolerance that's been used by doctors for the last 30 years that this hypothetical channel could have done and didn't, which is really upsetting, because if this person were successful, that would be a big deal. That would be a big deal. And it would be deserving of the millions of views and positive comments that video has. But if you aren't thinking like a scientist and aren't thinking about experimental design and thinking, maybe I should have a readout that isn't, bro, just trust me, I can eat pizza now. Like, it's frustrating because of the wasted potential and the potential dishonesty. I think being able to have scientific discussion is the basis of academia. People always discuss about each other's research. It's really fun too. Like, some of my favorite conversations is just people really into their own stuff and being good at communicating what makes that interesting, which is why, and I'm sorry to you programmers need to learn how to talk to normal people. Me talking to a biologist, no problem. Like, obviously, we're in the same field. Me talking to a chemist, also not really a big problem, because chemists, you know, it's a little harder, but they, in my experience, have been trained to talk to normal people. And my colleagues in the English department and the art department and the history department, they can all talk about their stuff in a way that is accessible. You spend 2 seconds talking to a neurocomputational biologist, and we shared a building with neurocomp people who use programming to think about the brain. Just instantly lost, no attempt at making the material accessible. I think that there is a dearth of practice with talking to people that don't have your background in those regions. Feel free to disagree with me, but that is my lived experience. What do you mean? You're all normal people, right? I'm talking to you all the time. I don't know. Not all of us are very normal, I think. If you're watching a Five the Neutrophil stream, you definitely are on the weird side, I would think. Nothing that would be that's not a bad thing, though. This is why technical writer is an actual profession. Yes, exactly. But the thing is that now that there's ChatGPT, that profession is under threat. But computer scientists still, I think, need training, formal training. They need to take a class, and every time that anytime to talk to a normal person, and the normal person goes, I have no idea what you're saying, they need to have points taken off until they learn the way to talk to normal people. Welcome to the Help with T-Cells. Thank you so much for subscribing, Aiden. Your support is very much appreciated. It's also really good to have more people in professional roles in biology, just because, like, part of my fear as a streamer is that I am going to eventually at some point propagate biology misinformation, and having more professional eyes on my content gives me a sense of relief that there are going to be people who can call me out before stuff gets too out of hand. So thank you so much for the membership. If I print hello, if else touch grass. Wouldn't be surprised. You are talking to a cartoon cell about genetics of schizophrenia. Of course, I'm very thankful for you all for being here, but yeah, and I don't know, shout out to the neurodivergent community too, because there have been quite a few comments on my videos that have introduced themselves as people who are neurodivergent, and I didn't know I had that appeal. But I'm very happy to have you in my listener base, all of you. It's very cool to see that my content does something for you in that kind of way. All right, okay. I think, for as much fun as this was, it is time to call this stream to an end. It's been so much fun talking about genetics. And again, I was really, really dreading until I came up with this schizophrenia story because I wanted a way to make it interesting beyond Punnett squares, because I don't think that's interesting. And I'm glad that you've, especially to y'all that have stayed to chat, because chatting is also very fun part of this process. But I'm glad you have all taken the time out of your day to hang out with me. I will see you next week where we talk about bioinformatics using computer powers to destroy bacteria that are drug resistant. Going to be very, very exciting. So see you all next time. Bye, y'all. Oh, Dr. Toto, just missed it. Bye, y'all. Nope, wrong screen. Bye, y'all.